Okay, so good afternoon, everyone. Very nice to have you here. I think it's a great opportunity to share some ideas, some results, and to make network between our colleagues from France and Brazil. When Professor Zago, Professor Márcio, and Leandro Colli idealized this kind of events, we thought that maybe this should be focused on the students. So this is made for you. So we hope that the students can, after each session, can make questions, because this is the best way that this can actually strengthen the collaboration and make you curious about science. So we're going to have the most important, as I was discussing with my French colleague before, the most important session of the whole conference is about immunology, and this will be done in this early afternoon. We have 4 speakers, 2 from Brazil, 2 from France. After that, I will ask them to sit here and answer the questions. And of course, again, if you want to have lunch, you should make a question. Okay, if not, you're going to stay here for a while. So my name is Niels Câmara. I'm a professor of immunology. That's why this is relevant here. And I'll be very happy to host this session. And I'd like to start inviting Professor Alexandre Keller. He's an associate professor at the Federal University of São Paulo. Alexandre, we are very, very, very happy to have you here. You have 20 minutes, a little bit more, no stress about that, to present your work, okay? So the point is here.
Thank you. Okay. Thanks, Neil, for presentation. Thanks, organizing, for inviting me and giving me the possibility and the opportunity to show our work. I'm going to talk about NKT cell biology, past, present, and perspectives. So this is my history with NKT cells, so what we can work with and maybe what we can do together. It will be like a brainstorm, so let's start that. They are non-conventional T lymphocytes because they have an invariant T cell receptor that respond to glycolipids within the context of CD&D molecule. It can be exogenous antigens such as alpha-Gausser, GSL1, PBS44 that induce a protease-1 response, OCH that induce a protease-2 response. They also can answer or respond to stimulation by the protein molecules at the membrane like FAS, NKG2D, OX40, NK1.1, and also to cytokines such as IL-12, 18, and IL-33 that are associated with infections and tissue damage. So they can be targeted by specific agonists, so you can modulate immune response with them, thinking about immunotherapy, and they also respond to danger. I mean, danger is stressed cells or cytokines associated with infection or damage, and then they can modulate physiology or pathology through the endogenous ligand that still is an obscure subject because we really don't know who they are. And these 2 topics are strictly related, so I will be oscillating between them during my presentation. So my history with NKT cell started a long time ago during my PhD with a part of PhD with Boris. Then I started to work with Marion during my postdoc. She was my supervisor. And where I get the chance to work with François in Lehuen. At this time, I work with Oncology, so we demonstrated the NKT17 subset and the thymic pathways that lead to the differentiation. Work a little with the response of invariant and non-invariant natural killer T cells to pathogens, the schistosoma, and the role of NKT cells during allergic inflammation and the modulation of anti-GBM glomerulonephritids and autoimmune disease. And this work marked my return to Brazil, where I was kindly welcomed by Niels, and he opened his lab. So I started my own research group. Our first work together was showing that using the GSL1, the protease-1 agonist, we can modulate the development of experimental focal and segmental glomerulosclerosis. That's a disease associated with a TH2 pattern. So yes, we can change the immune response and it can modulate disease. After that, I work with Vera, showing that NKT cells are important in the host resistance against fungal infection and also that we can improve this resistance using alpha-Gausser, another agonist that can activate CD8 cells and protect from fungal burden. And in this field also we did an experimental methodology to vectorize agonists in polymeric nanoparticles. Two things very interesting we had. First, we improved the effect of alpha-Gausser, so NKT cells were more powerful in the response because we increased the charge of alpha-Gausser in the surface of target cells, and we bypass the use of organic solvents. That's a problem when you use anti-lipids, so it was more, you can say, safety if you want to use it in humans. So this is part of our work about immunotherapy with NKT cells. Then I get curious about external factors that can impact NKT cell biology. I start this work with Daniela. So we work with a model of sleep deprivation, so we can block the REM phase of the sleep cycle. In these two works, we show that first, when we have allergic mice that develops eosinophilia and Th2 response, once they are subject to REM sleep deprivation, they go to a neutrophilic pattern. So for mild asthma to severe asthma using this model. So it's important to understand how the disease can progress. And also we show that sleep can compromise CFH differentiation and the production of neutralizing antibodies. So it damps adaptive response to malaria. This is the model that we use, a multi-platform modified model. So animals trained by 3 days in a dry cage. After that, we put the water just at the line, and every time they want to get in the REM cycle, they touch the water and wake up. That's what we have, a maximum for 3 days, because after that, they can survive, at least in our model. So the first question was I lost one, okay. If it can, how can I say, impair NKT cell response. So to induce NKT cell response, you give alpha-Gausser, then we pass them to the paradox of sleep deprivation model, activating alpha-Gausser, the NKT cells with alpha-Gausser, and no change. They respond as well as, so they are resistant to sleep-induced stress. It was very good at this time. So we know that stress can activate at least two axes, the HPA, glucocorticoid release, and the sympathetic nervous system with the release of norepinephrine. So we have adrenergic signaling. So the question was, how adrenergic signaling impact NKT cell response? Because we know that it inhibits a lot of immune response, such as anti-tumoral immune based by NK cells or NK cells and CD8 T cells. So if I want to treat with alpha-Gausser, and we know that during stress response, there's a lot of adrenergic signaling, may it impact negatively our treatment? So the question was, does NKT cell rescue NK or CD8 T cell activity during stress response based on adrenergic signaling? So we worked with Basso in this study using the other two AC mice KO. They don't have the alpha-2 AC receptor, so they are hyperstimulated on the base of the sympathetic nervous system, so they present high levels of norepinephrine, okay? So the question was, if we get this animal, we give them tumor, and we give him a treatment with alpha-Gausser, it will work or not? Yes, it worked very well. The NK response were okay. The CD8 not so good, but suppose that they are already resistant, no big deal. But the most interesting thing that we found here was that these animals were protected from the liver damage induced by alpha-Gausser administration. This is a collateral effect of activation of hepatic NKT cells. So no high levels of hepatic enzymes, no necrotic area, so they were protected. This was unexpected for us, okay? And the question was, can we use this data to modulate other kinds of disease or modulate somehow the activation of NKT cells or inhibit the liver lesion? So you used yohimbine, that's a pharmacological inhibition of the alpha-2 adrenergic receptors. It means to mimic the KO mice. And we had the same response. You used also PBS44, that's a powerful agonist that induced a more TH1 response. It was also the same profile. So from that, we think that we can use targets in adrenergic receptors to mitigate invariant natural killer T cells, acute liver injury. That was we're not expecting, but it appears in our hand. So the question now is, can we use the same strategy to control other inflammatory models, where we can use it in asthma, can use it in cancer or whatever? Let's see. We are working on it. After that, Pedro proposed to work with cell metabolism and NKT cell response. There are a lot of data produced here, but what took my attention was the fact that the glycolysis is essential for interferon gamma and IL-4 production upon NKT cell activation. So if you don't have glycolysis, we don't have interferon gamma, we don't have IL-4. And what was found that depends on the PKM2 activity. But here we had to pay attention because PKM2 activity can be both, means both metabolic activation, so induction to give fuel energy, and also transcriptional effect. So here we show that it's mainly glycolysis that is dependent for the activation of NKT cells. So my question now is, we are thinking about immunotherapy, induced cytotoxicity. We know already that PKM2 is important to induce activation of NK cells and CD8 cells if we think in the interferon gamma production. And what about the direct cytotoxic effect of NKT cells? We don't know if the activation of FAS, NKG2D, or perforin granzyme depends on the same pathway. Maybe someone will say, oh, of course, but we're talking about NKT cells. They don't like, of course. You have to test, because every time you think that is okay, to be sure to do that, it's not the truth. So why I'm talking about this approach? Someone talking about use alpha-Gausser as immunotherapy, NKT infusion, CAR NKT cells. But we must remember that we're talking about cancer cells. Cancer cells are high glycolytic cells. It means that they use a lot of glucose. So if I need NKT cell to use glucose, how can I work in a glucose-deprived environment? Also, we have hypoxia that increase glucose uptake also via the cancer cell. So if I have, again, another way to have glucose starvation, it probably will impair NKT cell activity. So this is one of our questions now. How can I boost the activity of PKM2? And then I was talking about Zeca. I have a problem because when it's saying about boost PKM2 means what? Metabolic pathway or transcriptional pathway? So my first question come back. I must know now if PKM2 is also responsible for the activation of cytotoxic front of the NKT cells. Well, this is the one thing that we are working now. Again, immunotherapy. We published last year some data showing that we can make some chemical derivation from RQS to have powerful compounds with anti-tumoral activity. So the question now was, can we use these compounds to improve alpha-Gausser activity or anti-tumoral efficiency? So question now, can compound one, this we have a lot, okay, increase the NKT cell response to alpha-Gausser? This is the first question. So you use an hybridoma and a B16F10 melanoma cells. They were charged with alpha-Gausser, treated with our compounds, and they put together. Then we look for IL-2 production. That is the first response of this kind of cell, of these NKT cells. But we found that, yeah, I have much more IL-2 when I compare with alpha-Gal ulcer itself. So somehow it is improving the response of NKT cells to alpha-Gal ulcer. Well, a long time ago, they said that antigen-specific cytotoxicity depends on Fas-Fasel. It is correlated with antigenic potency. So what does it mean? This cytotoxicity mediated by CD1D is Fas dependent. Okay, so I must have a high expression of CD1D, and also a higher load alpha-Gal ulcer in these molecules. And also, I must increase the expression or Fas or Fas-L or both. So, how was the expression after treatment in the B16F10? We increase the CD1D and also increase the Fas expression. So, two pathways that can activate NKT cells are increased using this compound. So, it can explain why it improved NKT cell response. So, but how it's working? That's the second question now. Some clues. It appears that HDAC controls CD1D expression. So, okay, if I improved and increased CD1D expression, maybe I am working with HDAC. So, we did the first assay using sodium butyrate as control, and yes, we decreased the expression of transcripts to HDAC1 and HDAC2. So it appears that our compound is working on stone desacetylase. But this is a very, very huge problem, because when we are talking about HDAC, we are talking also about metabolism. Then I come back to PKM2, all that stuff, and there are other things that may be like several membrane or protein complexes that can be regulated. And okay, we have a lot of trouble and work for some years, I think so. So we are looking for the mechanism that is acting here. Well, then we get back to stress, because although we worked with alpha-Gal ulcer, the question is, stress work in the same pathway in other models that not alpha-Gal ulcer? It means NKT cell response activated by something else than alpha-Gal ulcer is still a resistance to stress. Well, we use now concanavalin A model that get the animal, you inject con A, and after 14 hours, you look at the liver and the tissue damage. So sleep deprivation plus con A, and then we get to look for the liver, and no liver damage. They were all protected. So, oh, that's great. But if you look at this picture, what you have here, two axes. One is the sympathetic nervous system, and the other one is the HPA that produces cortisol. So, okay, maybe the overexpression of glucocorticoid after stress is blocking an NKT cell or whatever activity. Yes, we get that. We blocked glucocorticoid receptors, and the animals were worse. So, yes, this is the pathway, but we're not fast enough, because it was already showed by Hudak. And this is interesting, because I did my first work with stress because of him. He said that NKT cells express TIDE, this is a suppressor receptor after adrenergic signaling, without any proof of that, just receptors. And we saw not that the same way. But what he said, that when you have a glucocorticoid, it changes the pattern of the NKT cells for a more suppressive. So we lost our work here, but not at all, because I changed the model. So let's see what happens when we have an acute sleep deprivation. So it means not 3 days, only 24 hours. Okay, what I have here is that it aggravated the liver lesion. So although 72 hours of sleep deprivation can block disease or damage because of corticoids, the sleep deprivation for 24 hours or acute stress activates something, because when we look at the animals, NKT cells, there's nothing changing in frequency or number, but they are activated. So something happens in 24 hours when you don't sleep that activate NKT cells. So we are thinking in 2 or 3 pathways of activation, but one that started is what about microbiota, because we know that stress can change microbiota. We did some assays trying to find something different with next generation sequence. We have some difference, but we have to do it again to be sure. So it's in line with the idea that deep deprivation can induce alterations in microbiota. So what we think is that when you're at steady state, there are a normal loading of nutrients and metabolites to the liver. When you give CONA, you get gut leak, so some nutrients and also bacteria go through and get the inflammation. That's normal, that's the model. And we have sleep deprivation, you change the microbiome, and then when you do you give CONA, this leak happening again with a different microbiome, which impacts negatively, and inflammation will be worse. So it's the idea that intrahepatic microbes can program NKT cells. So what we have in mind, a lot of things, okay? You can see that here I put 4 topics, but we had inside each topic more 4 and more 4 and more 4. We want to use high-dimensional microscope and flow cytometry to achieve these questions, to assess these questions, the response. And for now, our strategy is to have a 5-year grant. We have already a lot of Brazilian and American collaborators, and we are open for French collaborators too, or who else want to work with us. I have a lot of thanks, but a lot of colleagues, researchers, students, they all know that they are in my heart and in my mind. So thank you, merci beaucoup à tous.
Merci, Keller. Okay, it's fine. So I'm very glad to invite, we're gonna do the 4 talks first and then we open for questions, okay? So I'm very glad to introduce our next speaker, is Professor Agnes Lehuen. She's the director of the Department of Endocrinology, Metabolism and Diabetes and head of the lab Immunology of Diabetes at Cochin Institut, Paris Descartes. Merci beaucoup, Agnès. C'était un plaisir. Tu peux ici.
Muito bem, muito obrigada, Niels, pelo convite, todo mundo. É um prazer estar com vocês. Eu espero que vai ser mais interação científica com vocês. Agora eu vou soltar em inglês. Mas é mais fácil para mim. Okay, look, it's really a pleasure to be here. And it's great to talk after Alexandre because I'm talking about the sister population. Donc, Alexandre already mentioned that they have some conventional T cell, you know, that's recognized by MHC molecule, presenting peptides. But this is what we call non-conventional T cells. Donc, you heard about the first cells that was really heavily studied, NKT, and now I work a lot with MAID cells for mucosal associated invariant T cells. Look, I will show you briefly a little bit of highlights of the different studies which can be of interest for you. Okay, donc what are MAID cells? Donc, a little bit like Alexandre showed for the NKT, they have an invariant TCR alpha chain. That means everybody here has exactly the same MAID population in human and mice. And this semi-invariant TCR recognizes a non-polymorphic molecule, which is MR1, for MHC rated 1 molecule instead of CD1D molecule. And the interesting feature as well of these cells is they recognize a bacterial ligand. You know, it's not as the NKT, which has many, many phospholipids and many ligands. Here, up to now, we know only 1 agonist molecule, which is derived from the riboflavin pathway in the bacteria. Look, if you have germ-free mice, there's no MAID cells. Okay, they really need the bacteria to have a presentation of this ligand by the MR1 molecule to activate the TCR. And in fact, it's a very smart way for the immune cells to recognize bacteria alive, you know, which produce this ligand and can activate MAID cells. Donc then the MAID cells have 2 major, a little bit like the NKT, they are like effector memory cells. That means they can be activated by both ways, either through the TCR on the left hand, from I just told you, the TCR recognizing MR1 presenting the bacterial ligand, But as well, like the effector memory cells, they have already a lot of cytokine receptors. Now, they can be activated through IL-18, IL-12, for example, but as well type 1 interferons and so on, IL-15 and so on. And once they get activated, they can have different functions. They can be cytotoxic by producing perforin and granzyme B. They can, of course, proliferate. And then they can produce various cytokines, which are rather pro-inflammatory, like TNF, interferon gamma, IL-17. And as well, they can also produce chemokine, which will attract monocytes and promote the differentiation in macrophages. Now, the first study I will mention to you briefly is a study we did with the children who have type 1 diabetes. So type 1 diabetes is an autoimmune disease when the immune system kills beta cells. There's no insulin produced, and in fact, the children, nobody with diabetes can live when it's the autoimmune disease, which is very severe, and they need injection of insulin. And when we analyze these children, you can see on the left the frequency of the made cells in the blood. Donc in blue are the controls. In red, the kids at the onset of the disease when they get at the hospital, because it's very severe in the young children. Often they go up to the intensive care, and they need insulin treatment, and then they, of course, feel better. And when we analyze various markers, just for simplification, I'll show you the activation marker CD25. You can see again in red that the kids at the onset of the disease, they have fewer mates, but the mate cells are more activated. And when we look at different functions, what was the most exciting was the production of granzyme B. These cells can be highly cytotoxic, already mentioned that to you. And again, what was very interesting to us is you can see in the kids at the onset, look, we used to say type 1 diabetes is like a teenager disease, but it's not true anymore. It's really very young children. In fact, many of the young kids, like 2 years of age, can have type 1 diabetes. And in fact, these very young kids, which have a much more aggressive disease, is why they get it much earlier, you can see that the male cells are producing much more granzyme B, suggesting that by the cytotoxic function, the male cell can participate to the killing of the beta cells. And then, of course, to address this question, we have to move to mouse model. And I tried in my studies always to combine both the studies in human with mouse models. And here you can see the schematic view of what is happening. I don't think I have, okay, maybe here, do you see? No. You think it's work here? That's okay. I hope the slide is clear enough and you can get it. On the center, you can see the pancreas. And when we have a mouse model, this mice develop type 1 diabetes spontaneously because they have a genetic background to develop the disease spontaneously. Now, with NOD mice, when the disease evolves, there is a recruitment of the made cells in the pancreas, and these made cells produce more and more granzyme B, and they can kill beta cells. But, okay, perfect. But what was interesting in the mice is that we can study all tissues. And when we analyze the gut, because remember, made cells stand for mucosal-associated invariant T cells, are quite abundant in the gut mucosa. And in the gut mucosa here, they produce other effector molecules, and mainly IL-22 and IL-17. And these two cytokines are key to maintain the gut integrity. In fact, the two cytokines act on the epithelial cells. They allow the production of mucus, they allow also the tight junction protein, and they can maintain the gut integrity. However, when we did MR1 mice that don't have any made cells, then you have a linkage of a gut barrier. You have a translocation of bacterial component, for example, RPS, bacterial DNA, peptidoglycan, and these activate the immune cells in the pancreatic lymph nodes because the pancreas is very close to the gut. And in fact, when you have this translocation of bacterial ligand, you can activate dendritic cell, which favor the development of this autoimmune reaction. Look, in other words, the made cells has two functions which are totally opposite. There are bad players in the pancreas, they can kill beta cells, but they are key players to maintain the gut integrity. And in fact, it's interesting to see what is the most relevant. And when we analyze this MR1 deficient mice, in fact, the mice has more diabetes, which showed that the protective role of made cells in the gut is much more important as compared to the killing of the pancreatic beta cells by a made cell. Then the thing, of course, which is frustrating to study type 1 diabetes, we don't have a target tissue in human. You cannot study much pancreas. Look, I turn on as well to the type 2 diabetes and obesity when I can study adipose tissue. And here, what you can see is when we have this code, when this is adult patients, these are the controls, healthy controls. The patient with type 2 diabetes has, again, about 10-fold decrease of the MAD cells in the blood. But when we analyze the obese patients, you can see that they have a major decrease of the MAD cells in the blood, and in fact, about 20% of them do not have any MAD cell detectable. And then, again, we analyze various markers, and for simplification, I just show you CD25, and again, MAD cells which are very fewer, but the few which remain are much more activated as compared to the MAD in the healthy control. Look, what was interesting, I just mentioned that to you, is I wanted really to study the target tissue, which in the context of obesity is adipose tissue. And you can see when we compare the blood per sample from the same patient, the blood and the adipose tissue, you can see the patient that have a lower frequency or even no MAD cell detectable in the blood, whether one will have a higher frequencies of MAD cells in the adipose tissue, who does suggest that with the recruitment of the MAD cells from the blood to the targeted tissue. And in fact, this is something which is very common to all inflammatory status autoimmune diseases, with a decrease of the frequency in the blood and a recruitment in the target tissue. And when we analyze the function here, what was striking was the production of IL-17. And you can see that indeed the MAD cells in the adipose tissue produce very large amount of IL-17 to some level which have never been described up to now. And this is much higher as compared to the conventional CD4, CD8, or the gamma delta, which here is written as double negative cells. And interestingly, in fact, further studies really suggest that IL-17 is a key molecule to induce insulin resistance in the adipocytes. And another interesting feature of these studies is I was fortunate to work with Karine Clément at La Pitié-Salpêtrière, and she has a huge cohort of patients. We went on for bariatric surgery, which is nowadays one of the major procedures to avoid comorbidities of these patients who are severe obese. And indeed, here you can see when we have a patient 3 months, 6 months, or 1 year after the surgery, with a very nice decrease of a body mass index. As expected, they do lose weight. And nicely again, the patient with a diabetic, which have a higher level of glycated hemoglobin, you can see that the level will decrease again after the surgery. And when we analyze the MAD cells, these are the patients before the surgery, with some having no MAD cells at all detectable in the blood. You can see 3 months after the surgery, they almost come back to the normal level. And this was even a little bit nicer, 6 months and 1 year after the surgery. And I think these data are quite interesting and fascinating. That means that, in fact, there's a strong link between the metabolism of the patients and the frequency of MAD cells. And maybe you're aware that many of patients with type 2 and obesity are very susceptible to infection, particularly bacterial infection. Look, it's very nice that if after treatment, they have improved metabolism recovery, and they as well can find back the frequency of MAD cells, which are very efficient to fight against infection. And this, again, I will just summarize on one slide all the work within the mouse model to understand the mechanism. What's happened when we put B6 mice under high-fat diet, but they become obese? The MAD cells become activated. They produce 17 and TNF-alpha. And the adipose tissue is quite inflamed with a whole modification of the immune cells environment with more M1 cells, more CD8, and we lose some regulatory cells like the Treg cells, here shown with eosinophil and so on. And here, interestingly, again, when we analyze the gut mucosa, because MAD cells, again, are very abundant in the gut mucosa, we could see that MAD cells in the context of obesity promote the inflammation of the ileum. They promote the dysbiosis, which has exactly the same type of bacteria increase as in patients. And with more inflammation, dysbiosis, and always as increased gut permeability, which again is already known that the translocation of LPS is a key component to promote inflammation, like we call the chronic low base inflammation observed in obesity, which can again promote the insulin resistance. Donc together with the local inflammation in the adipose tissue and the release of LPS, with a major inflammation, insulin resistance, and the development of type 2 diabetes. And at the time, I will not show you the data, it's in the paper if you're interested, but we demonstrated a very strong crosstalk between macrophages and MAD cells. And what was very nice, if we use a small molecule, which is an antagonist, the acetyl-cis-FP, which is in fact is derived from vitamin B9, this small molecule can bind the MR1 molecule, but block the interaction with the TCR of the made cells. And when we treat the mice with, they were already obese, so we had obese mice, we treat with this inhibitory molecule, and we dampen the inflammation, and we can restore a good metabolism. So this is kind of very exciting, but with a small molecule, we can reverse obesity, and we can reverse the metabolic dysfunction. Look, let's hope maybe one day can also work in human. Look, when we're doing all these studies on type 1 and type 2 diabetes, like everybody else, and I'm sure unfortunately in Brazil, but we were very badly hurt in Paris with a COVID infection. And since we're working in other studies I will show you later in the mice with infection, we decided to really participate to this effort, because at the time of the beginning of the COVID, it was about 40% of the patients in intensive care units who passed away. Okay, so let's see what the made cells are doing. And on the top panels here, I will show you the activation status of the made cells in the blood. This is the marker CD69, which is an early activation marker. In green, you have a control cohort with some diabetic, because you are aware that more diabetic patients are hospitalized and went in intensive care. Now, this is the proper control cohort. In blue are the patients who were in the infectious department, that means they have a more moderate disease. And in red, the patients who are in intensive care unit. And you can see that the activation status of the made cell is really amazing. I mean, some of them, all of them get very high level of CD69. Again, never seen before in any patients. We as well analyze CD56, because this molecule as well is associated with the activation of made cells, and even more from the cytotoxicity function. Look, when we look with this expression of these two molecules, it's much more increased in the patient intensive care unit, with a patient in the infectious department, and here the controls. But this is the non-supervised analysis. We again show the patients in red are here on the bottom, the high expression of CD56 and the high expression of CD69. And since we're in the context of a virus infection, we analyze all the cytokines. But interestingly, the granzyme B again stand out when we compare the controls individual here, the patient in the infectious department, and here the patient with a severe disease. And here, it's different from the other studies. This is, we take the cell directly from the blood, and they do produce granzyme B. We don't have to do any PMA or nomycin stimulation. That means these cells are highly cytotoxic in the blood of these patients. Then, of course, what was important is to try to have, again, the target tissue, which is in the COVID, is mostly the lung, what is the main target for the survival of the patients. Look, we have pair samples from the blood and the lung fluid. Look, in this case, is the endotracheal aspirate, because when the patients are in intensive care unit, they cannot breathe anymore. In fact, they need to have the tubes and to have the fluid removed, but they can have a contact with the oxygen in the lung. But this is really ethical to have this lung fluid. And you can see again that in the target tissue, which is the lung, there is more made cells as compared to the blood of the same individual. And when we look at the function, the cytotoxicity, the made cells are much more cytotoxic in the lung as compared to the blood. A little bit with what we show for obesity, the effective function is always much stronger in the targeted tissue, which is the lung here. Look, what we think, and I will show you a bit more data then, is why the made cells are linked to the mortality of the patient. Look, I will first show you the data on the comparison of the mortality. In this case, it's the activation status of the made cells in the control, patient with moderate disease, patient with a severe disease, and the patient who passed away, because I told you about 40% of the patient passed away. And you can see that the activation status are the highest. And it's much higher as compared to the other immune cells we could study in the blood, conventional CD8, gamma delta T cell, NK cells, and CD4. Look, the made cells seem to stand out among the other immune cell population. And when we look in this diagram, what you can see is everything which is behind this red cycle. That means significant differences for the patient who died as compared to the patient who survived. And the major parameters you can see here, as 4 parameters of the made cells: production of interferon gamma, granzyme B after stimulation, spontaneous granzyme B, TNF. No other differences with the other immune cells except the gamma delta T cell. And interestingly, in this context, the gamma delta T cells are producing IL-10, who shows that even though there are also non-conventional T cells, the gamma delta T cell try to dampen the inflammation. Look, a very different function as compared to the made cells. Then we did as well some study in vitro. We had the chance to work with the SARS-2 virus in Bichat Hospital. And you can see again, when we compare all the population for the PBMC, when we put them in contact with infected macrophages, the made cells again are the cells which are the more activated as compared to other immune cells. And when we had a good batch of virus, we can even get in 3 different healthy donors a major activation of the made cells and a major degranulation, that means they're really cytotoxic when they're in contact with the infected macrophages, and they produce a granzyme B. But what was very surprising, and again a dogma, is we could dampen the cytotoxicity of the made cells when we use a non-blocking anti-MR1 antibody. Because remember, I told you the main agonist is a bacterial ligand, and up to now, in the influenza context, it's mentioned that the MR1 molecule don't have any role to play. There is no interaction between the TCR and MR1, which is important in the flu infection. In the context of COVID, it seems that MR1 is very important. And this brings me to this last slide for the COVID. In fact, in the non-infected patients, there's a good frequency of made cells, and they are not cytotoxic. In patients in the infectious department, with fewer made cells and they are cytotoxic. But in the patient in intensive care unit, there is even less made cells in the blood, they are more cytotoxic, but in the lung fluid, that even much more cytotoxic. And this was associated, it's shown in the paper, to a shift from the type 1 interferon response, which is protective in the context of viral infection, to the inflammasome response with L18. And in fact, the patient who passed away had very high level of L18 as compared to the patient who survived, which are more type 1 interferon and lower level of L18. But what was so striking to me, and I was trying to understand why the made cells are so much associated to the mortality as compared to other cells. And you have to remember, or to keep in mind, that often when patients died, they are not infected anymore. Maybe they don't have virus. That means that the specific CD8 T cells, which recognize the peptides from the virus, might not be efficient and very limited number. But the made cells are very numerous. I mentioned to you, they are recruited in the inflammation. They recognize MR1, which is ubiquitous. The all cells has MR1. And all our data showed before in different contexts, but upon inflammation, MR1 is really expressed on the surface. Because MR1, if the TCR, the TCR for made cells see MR1, they can be really killing. That means you have a major bystander tissue damage that can be caused by MR1. And this one, most of the time, in fact, MR1 is only in the cytoplasm. MR1 is never presented on the surface, except if you have a ligand. Look, in our context, you have inflammation, maybe some ligand from the lung microbiota, we don't know. But it seems that the made cells might be a major player by killing and making a lung tissue lesion. Now, briefly, in a few slides, I will show you some study we are doing in type 1 diabetes and Coxsackie. Because in fact, I work most of my time with type 1 and type 2 diabetes, and we're working on the role of viral infection in type 1 diabetes. Because often there's an autoimmune disease, we say, okay, what is the triggering agent? Because with HLA susceptibility, there's many other polymorphism, but why some patients, like the twins, some have diabetes, some don't have diabetes. And with many, many studies, and mainly by the Finnish groups, because the Finnish have a high incidence of type 1 diabetes in the world, and it's a smaller populations, they have wonderful cohort when they really follow the kids from the birth through the age. And they find that the presence of antibodies against Coxsackie or Coxsackie in the stool is associated with the development of type 1 diabetes. And now we have very nice bank because, unfortunately, of course, some patients with type 1 diabetes died, either from diabetes itself or from car accident or whatever. And when they found that there's always a presence of some viral proteins or RNA, it's an RNA virus, Coxsackie, it's an enterovirus, in the pancreas. Look, what we did, and we have done some study before on that, is if you take the mice, and when the mice is already pre-diabetic, that means there's a very strong infiltrate already in the pancreas, if we infect with Coxsackie, they develop diabetes in 10 days. So it's what we think might be happening in the children. And again, you can see here, when we infected the transgenic mice, which has a very high frequency of made cells, you have a very good incidence of diabetes. If we infect a MR1 without no MAID cells, you don't have a good induction of type 1 diabetes. Really saying again that the MAID cells might contribute to the killing of the beta cells and to development of type 1 diabetes. And excited about how study on type 2 diabetes and mentioned, on obesity, and with the work on COVID, we said, okay, let's try again. If we block the TCR interaction with the MR1, maybe we can block the development of diabetes. And this is exactly what we see. And again, it's supposedly the dogma, that MAID cells in the virus will respond to IL-12 and they will be activated. But this is really in vivo data, and this we have done for 2 years. Many, many experiments, transgenics, the nodes, and it's beautiful. It's really TCR dependent. Now, just the last summary slide for this model. I will not show you all the data, but we are almost, everything is completed. When we infect a prediabetic endodermis, when there's already a lot of infiltrate in the pancreas, the MAID cells get activated, and normally there's a lot of M1 macrophages, a lot of anti-islet CD8 cells, and they kill the pancreatic beta cells. But if we treat at the same time of infection the mice with this inhibitory molecule, acetyl-6-SP, which block the TCR interaction with MR1, now there's a totally change of the transcriptome, and they become like regulatory T cell. If you go in the data bank, they have exactly the same profiles of bona fide Treg cell FOXP3. But the MAID cells never express FOXP3, even in this context, okay? But we heard a very nice talk this morning with CD39, and this is one of the molecules which is the more expressed in the context of inhibition with acetyl-6-FP. We dampen the CCNI expression, we dampen the production of inflammatory molecule, and again, we have also an interesting molecule, CD200R1, which is also a very inhibitory molecule, which is upregulated. And it switched the F1 to M2 macrophages with much less inflammatory cells in the pancreas. And these cells now are not efficient anymore. They don't produce the granzyme B, an inflammatory molecule, which indeed can prevent the death of the beta cells. Look, for me, it's like one of the most exciting work we are doing is the role of this interaction with MR1 and the possibility to induce Treg cell, which of course can be so important in many, many contexts. And just the very final slide, I have been very lucky to work with a wonderful person. Look, I'll show to you today some work on the type 1 diabetes and the role of MAID in the pancreas, the role of MAID cells in the adipose tissue, the role of MAID cell, which can be protective in the context of type 1 diabetes, but might be more deleterious in the context of obesity, which makes sense as well, because we have a high-fat diet, and the first tissue to be modified, altered, is the gut mucosa. We have done some very nice studies showing that MAID cells can promote liver fibrosis, and as well the steatohepatitis. We have some nice work on Verlang already showed with the COVID data, and another study on the systemic sclerosis. It was a nice collaboration with Karine Clément when we showed that, in fact, the patients with obesity, severe obesity, which are diabetics, the one have a stroke, other ones have a lower level of MAID cells. And of course, one of my high interests now is to study the link between the microbiota, because it's a microbial ligand, and the activation and the function of MAID cell. A nice study with Renato Montero on lupus, and the link, it's a small paper, but it's very nice. It's a small cohort which has been followed after the treatment, and the patient will have a lower frequency of MAID cell. And the more MAID cell which produce granzyme B, they don't respond to the treatment, and they remain with kidney failure. And of course, some work with arthritis and many other studies. So if you have some ideas, I can provide advice, and we'll be happy to do that. And of course, it's key to mention the people who did the work. And this is the team, or at least some of the members of the team. And it's a nice place. You can come to see us. It's at the top of the Luxembourg Park in Paris. And this is in the cluster of Cochin Institute, because this is the cluster where Pascal, the philosopher, wrote his thought. Look, you might be inspired there. Of course, I need to mention my collaborators, and mainly Renato. I didn't put the name of the different collaborators, but the COVID studies was a lot done by Bisha as well. Work on diabetes, on MASH, and so on. Thank you for your attention.
Thank you, Agnes. That's why immunology is the most important session in this scenario here. After we have immuno-oncology, immunoneurology tomorrow, but today is just immunology. So now I'd like to invite our next speaker is Dr. Rodrigo Nalio. He's an immunologist. He works at the Instituto and also at the Faculty of Medicine at the University of São Paulo. Thank you very much.
Good afternoon, everybody. So it's a pleasure to me to be here today for many reasons. I will change a little bit the subject. I will talk about the macrophage subset that we described some years ago and the current projects we have in the lab. But first of all, before really start, I want to do a commentary about myself. So I'm a live result about FAPESP investment because I was in Christoph Koh's lab during my PhD. So it became a co-tutel thesis with Professor Barbuto and Dr. Christoph Koh. And we have some publications. And also Nathalie was my supervisor from PhD. So for me, it's a great pleasure to be in the same audience 12 years ago here. Okay. So as I mentioned to you, I'm mostly interested in macrophage biology and how those macrophages may interact with tumor cells and other immune cells in the tumor microenvironment. This review is very, very special because it's dividing macrophage subtypes and in more functional point of view. And so we do have some macrophages that are linked to the maintenance of homeostasis. We have part of those macrophages that also are anti-tumoral macrophages promoting phagocytosis and activation of some immune cells. And we also have probably the most known profile are the macrophages pro-tumoral that can promote many aspects like immunosuppression, angiogenesis, and so on. And so not so happy to be in Lyon for 2 years. I come back to Paris again for the postdoc. And so I spent more 5 years in Institut, Curie, and in Cordelier. And then there we started to go a little bit deeper in macrophage biology using single-cell RNA sequencing. So this paper we published some years ago. We have access of breast cancer patients' samples from luminal A, B untreated patients. We perform some single-cell RNA sequencing, and we uncover actually a subset of macrophage expressing this folate receptor, folate 2 or beta, if you wanna. And so we discovering many aspects of those macrophages. One is that those macrophages, if you look at the protein expression, we can stratify patients on folate 2 high and low. And patients that express higher amounts of folate 2 positive cells are the patients with the better outcome in luminal breast cancers. Then we went out to investigate how those macrophages could be spread in the tissue. And so here we used a series of different slides from breast and other types of tumors. And you may see, for example, here, probably you can see, yes, in red, those folate 2 positive cells are mostly located at the stroma of those tissues. And the other subtype, the TRIM2-like cells that are mostly related to monocyte-derived macrophages, they are mostly close to the tumor mass. We could quantify the proportion of those cells. We can see that in the stroma there is no difference, but we can see much more TRIM2 positive cells in the nests of those tumors. We also measure the distance of those macrophages to the tumor nest, and we can see that actually TRIM2 macrophages are more closed and folate 2 macrophages are more spread or far from the tumor mass. We then probably started to think about the functional aspects of those macrophages. We come back to patient data, and here using some signatures that we defined by single-cell RNA sequencing, we correlated the signatures of those macrophages, both macrophage subtypes, with different signatures of cells that are involved in anti-tumor immunity. And you may see here that folate 2 itself, the gene or the signature based on these 3 genes, they're all correlated with signatures for CD8, TLS, cytotoxic cells, T cells, in contrast to the TRIM2-like that do not really correlate. We also use some images to see that those cells are actually interacting with CD8 positive cells. We perform some beautiful images with Emmanuel Donadieu in the Institut Cochin, exactly. And we saw that those folate 2 macrophages may really interact with T cells, and T cells lose the capacity to migrate. They really interact with those macrophages, and they stay interacting very stably with those cells. We go so to the model, animal model. This is a very classical one, PYMT model. We did a very simple experiment. It's just sorting folate 2 macrophages or the other subsets of macrophages. We incubated with a peptide, and we put in contact with OT1 CD8, so the cells that really recognize peptides. And what we saw is actually those folate 2 macrophages are really able to induce the production of enzyme B, interferon gamma, or other cytokines compared to the cadm1 or the TRIM2-like macrophages. And we can see also that the folate 2 positive cells, they induce a more polyfunctional profile of those CD8 here by the co-production of 3 or 4 cytokines. But the most important part of this is that actually when he took a look and the folate 2 isolated from the healthy mammary gland, so without tumors, they do not induce some kind of T cell stimulation. They are not programmed to that. And so we went out in the mice, we isolated the folate 2 or the cadm1 macrophages from different tumors that have different sizes. So we have here for the healthy mammary gland, we have small tumors and have medium tumors. And we performed bulk RNA sequencing, and we saw that actually there is a kind of changing in the profile of those macrophages when the tumors start to be bigger. We investigated what kind of signatures those macrophages may express. And more for our surprise is that folate 2 macrophages in healthy mammary gland, they have a lot of genes related to metabolic process, so they are mostly in this phase of maintenance of homeostasis. But when you have tumor appearing, you have more genes related to leukocyte activation, immune cell stimulation, and etc. So for me, it was the most beautiful part of the work, because actually we can conclude more or less that those folate 2 resident macrophages are probably reprogrammed during tumor growth. And so my main question was, so there is some kind of impact on patients' outcome. And so in this time, I took a flight back to Brazil 3 years ago, and now associated to the 2 institutes, the Faculty of Medicine from São Paulo, from USP from São Paulo, and IDOR, to answer some questions more focused on those macrophage biology. I will just put some information about IDOR, because I don't know if everybody knows about our institute. So IDOR, we have many institutes, many filials in different states of the country. We have the 4 main states in Rio and São Paulo, Bahia, and Federal District, that we do research projects. We have clinical and translational research programs, and we have also undergrad and graduate programs. This IDOR is one arm of the HEDIDOR. It's a network of a lot of hospitals. And so it gives us a lot of important numbers. So we are now more than 150 PhDs among researchers, teachers, and collaborators. We also have more than 100 researchers that make part of the whole group of researchers. We produce more than 2,200 papers, and we collaborate a lot with different countries because they're very, very collaborative in there. And so probably linked to my presentation, we have the main areas of interest from IDOR is oncoimmunology or immunology and oncology separately, but those 6 subtypes of fields are the most important there. And we have access to a huge number of patients because of the number of hospitals we have. So we calculate about 10% of all the oncologic patients we have in Brazil, they are in the network. So it's a huge amount of quantity. So we have access to many types of samples. And it's why when I come back to Brazil, I started to think about the new projects I can develop here. And one of those was focused on colorectal cancer. First, because I was in Jérôme's Gallon lab, so I had all this information about colorectal and T cells. And the second one is because colorectal cancer has become one of the most important cancers nowadays. We have also a very increasing number of young adults developing colorectal cancers. It's a huge problem of health problem from different countries, Brazil is included. And so you may know the works from Jérôme Gallon. They use this kind of score using the expression of CD3 and CD8 markers, looking at the density of those cells in the tumor margin or in the core of the tumors. And they publish a lot of papers, but what is important, as I mentioned, is that when these patients have a higher immune score, meaning more CD3 and more CD8 in the margin or in the center, those patients have the most important curve of survival. So it was proved for many, many patients in colorectal cancer. And so we continue to collaborate with them, and we have a cohort, a French cohort that we started to work when I was in the postdoc, that we performed many types of experiments using the same cohorts. And our main question at the time was how the immune cell landscape of these patients, including the folder 2 macrophages and TRIM2 macrophages, and how those cells may probably crosstalk. So starting by the immune score, here we have all the patients. We have the expression of CD3 and CD8 in the center of the tumors or in the invasive margin. You may see that are very, very heterogeneous depending on the patient you look at. So we decided to divide those patients by the median, so taking the higher and the lower expression of those CD3 and CD8 positive cells. And so we perform a very simple analysis using the bulk RNA sequencing from the same patients, looking at what kind of genes could be common between patients expressing higher levels of CD3, CD8 or lower levels of CD3, CD8. So what is interesting, when we look at the tumor margin on a tumor center, and you look at higher CD8 and higher CD3 in both zones, we saw, surprisingly, the presence of some genes that we know before. So folder 2 was included among this. So when you have higher frequency of CD8 and CD3, we have the signatures of folder 2-like macrophages appearing on that. So we come back to the tissues and we perform a multiplex images using this panel, looking at mostly monocytes and macrophages and T cells. And what we did now was to separate in different zones. So I have at least three zones of tumor, the center of the tumor, 3 in the tumor margin, 3 in the normal adjacent tumor, and 3 in the stroma. And we could compare the density of different macrophage subsets and T cells on those patients. One thing that's very interesting is that depending on the zone that we look at, those different kind of cells, we may see that there is a changing in the frequency of those macrophages when you go towards the tumor margin or tumor center. So meaning when you go to the tumor margin, you have a decrease in the proportion of folder 2 positive cells and an increase of CD14 and TRIM2 positive cells. But we still keep having a lot of different kind of cells in both zones, so the tumor margin and tumor center. And so what we did was we come back to the tumor, to the immune score, and we dividing those patients and higher or lower for CD3, we may see that patients expressing higher levels of CD3 in both margin and tumor center also have a lot higher expression or higher density of folder 2 positive cells. And we saw actually that those cells in colorectal cancer is much more heterogeneous. So we may have folder 2 expressing also TRIM2, CD14, or the two at the same time. It's much more complex how those cells may differentiate between each other, and we want to understand a little bit how it's working. And so my present project here is, so if we have some different changes between those folder 2 macrophages that have this profiling of metabolic towards a profiling of immunity modulatory cells, I want to know how, when, and where those cells may be modified or may be sensitized by the tumor products or the tumor microenvironment. And we have two main questions. One is probably those cells could be used like immunothermometers. So if they are in the tissue, they are tissue resident before the tumor appearance, probably they will feel very, very rapidly how those little mutations can induce tissue differentiation. So this is one question. Another question is probably the interaction of those macrophages with T cells in the different zones that we want to look at may probably be predictive of the early cancer onset. Those are the major questions we have in the project. And to answer those questions, we have first an animal model that we are developing in collaboration with Professor Niels and Professor Vinicius. And so this is a very classical model for colitis-induced cancer that we injected OAM in the beginning, and then we have cycles of DSS to induce inflammation. And we do set up experiments to do colonoscopy in those mice, and so we can see the appearance of some polyps at day 38, more or less. We can also see by histology the appearance of some hyperplasia in these mice. And we also set up some experiments using multi-color flow cytometry to really see our FOLD2 positive cells. So one big idea of this is to collaborate. We are already collaborating with Julie Elft and in France, and they developed the FOLD2 mice, and so we will probably use the system to deplete or to downregulate some kind of genes. And so to answer also in a more translational way, we also have access to different tissue patients, and here we have access to different types of polyps, low risk and high risk polyps. We have in situ tissue tumors, and we have T1 to T4. So our main idea is to use those tissues in a more spatial temporal profiling, and so we can stain for different kind of cells, and we can use digital spatial profiling from NanoString. It's the first one we have here in Latin America to really question about the molecular profiling of those macrophages in these different zones and in these different patients' tissues. And so our main idea is to reconstruct in a time spatial dependent how those macrophages may behave during tumor development. This is the main idea. And so we have some preliminary data on that. I didn't know that we were in YouTube, but I put, we performed some analysis. So here is one slide of the patient. And so we had different zones that we were focused. Here, the non-tumoral. I don't know if you may see here, we selected some zones in the different tissues. We have the intratumoral zone. We have the stromal zone, very far from the margin, and we have some margins. And so what we did actually, we collected mRNA only from FOLD2 positive macrophages. So we stained the slides, and we also check it for double, triple positive using vessels or CD3, and we only collected the mRNA from the FOLD2 positive mice. FOLD2 positive macrophages. So I passed very fast on that. So here it's an intratumoral port. You can see here the vessels stained for CD31. We have some CD3 positive cells, and we have our FOLD2 macrophages. And here is how we capture the mRNA. So in this slide we have 21 cells that we can capture in sequence. We have in the tumor margin a little bit more cells, 88 cells. We have in the non-tumoral part, they have much more of those macrophages. Here we cover 131 cells. And we also have from the stroma. And we perform a very simple analysis. It's just we compare the genes that are differentially expressed in between those 4 zones. And what we found, sorry because I didn't put the name of the genes, but what we found is actually that depending on the zone, you look at those macrophages, there are modules of genes that are upregulated and downregulated. And so our idea is now to perform a more broad analysis using polyps and established tumors, and also correlate this with the mechanistic tools in mice, trying to answer the question about how those macrophages may crosstalk with the tumor microenvironment. And so this is the team, so the spatial team, so they are all motivated to use this first type of experiment. So all the people that helped me to collaborate, and the IDOR, the pathology, molecular pathology from Regidor, and some professors that are giving a lot of support. And here, all the other external collaborators, and here we still collaborate with people from France. We still collaborate with many colleagues here, including Professor Niels and Thiago is here. And I just put here that we have 2 grants waiting response, and so we cross the fingers to have a very fast answer and to continue this work. So thank you very much for your time.
you, Rodrigo. I was a little bit scared that he went too far on oncology, but it's still immunology. So thank you very much. So now it's my pleasure to invite Dr. Nathalie Bendriss. She's a group leader at the Cancer Research Center at Lyon. It's very pleasure to have you here. Merci beaucoup.
Thank you very much for the introduction. So I'm very pleased to be here today and to be for the first time in Brazil. And I am very proud also to see what Rodrigo is becoming. And I think it's a very good example of fruitful collaboration between France and Brazil that was initiated thanks to the FAPESP fundings for the PhD sandwich of Rodrigo when he joined the lab in 2012. So I am immunologist, but applied in oncology. So I'm sorry, but maybe I make the transition with the next session. So I will discuss today with you about the dynamics of innate immune cells across tumorigenesis, which is a program that I developed within the team directed by Christophe Co at the Cancer Research Center in Lyon, which is hosted by the hospital Centre Léon Bérard, which is one of the most important cancer centers for patients in France. So you probably all know that immunotherapy has really revolutionized the treatment of patients, especially thanks to the approval of anti-PD-1 and anti-PD-L1 in oncology. It means molecules that will mostly target adaptive immune response and in particular T cells. Nevertheless, as you can appreciate on this graph, the efficacy of the anti-PD-1, PD-L1 in oncology, even though if it works for some patients, is still limited because most of the tumors or the patients with some types of tumors do not respond so well to the anti-PD-1, anti-PD-L1 immunotherapy. So we strongly believe that there are new challenges in immunotherapy, and in particular, we believe that in the immunotherapy-sensitive patients, it is probably more important to treat the patient earlier, and it is evidenced by the recent results of clinical trials in stage 3, for example, melanoma, where patients respond more than stage 4, but also with the results from the neoadjuvant treatment compared to the adjuvant treatment. So we are thinking that it's maybe at the era of immunointerception and immunoprevention to improve the efficacy of anti-PD-1, anti-PD-L1. Another important aspect is that, as I mentioned earlier, there are a lot of resistant or refractory patients to immunotherapy, and so it is very important to define new combinations and to identify novel immunotherapy targets. And in this context, in the lab, we strongly believe that it could be important to target the other arm of the immune system, and in particular the innate immunity, to strengthen the therapeutic arsenal based on the adaptive immune arm. So in the lab, we have been interested for many years in understanding the interplay, the complex interplay between immune cells and tumor cells. And for many years, we have been mainly focusing on what happens in advanced tumors and what are the mechanisms involved in the immune escape. And in particular, this work leads to a lot of publications. And more importantly, we describe two main pathways that leads to target validation and patent that has been licensed, for example, to GSK, with one antibody targeting ICOS, a molecule that is involved in the crosstalk between a subtype of dendritic cells and regulatory T cells, and that is important to maintain an immunosuppressive network in these advanced tumors. And another good example is the work that Rodrigo developed in the lab when he joined in 2012, where he demonstrated it characterized the macrophages in the advanced tumor and see that the microenvironment sensitized the monocyte in the periphery that will lead to the development of immunosuppressive macrophages. But more recently, we decided to move a little bit forward and to change the project we developed in the team. And in particular, we are now mostly focused on the mechanisms that are involved in the surveillance of the tumor cells or the tumor initiation by the immune system. And in this context, we are experts in three main populations, or yes, the macrophages, the NK cells, and the dendritic cells. So what are the dendritic cells? They are rare immune cells, but they are very important to initiate and orchestrate immune responses, in particular because they are the ones that are specific to present antigen to naive T cells and generate effector T cell responses that will be important to fight against tumors. But this is not so simple because the family is very heterogeneous and there are different subtypes of dendritic cells. And in particular in the lab, we have been interested for many years in understanding the role of plasmacytoid dendritic cells and the CDC1 dendritic cells. So these two populations are clearly different in terms of phenotype, but also in terms of function. In particular, the plasmacytoid DC are specialized in the production of type 1 interferon, while the CDC1 are specialized in the production of IL-12 and type 3 interferon. And it has clearly been demonstrated that the CDC1 are the ones that cross-present the antigen and initiate the best CD8 anti-tumor specific immune responses, while the PDC are more innate cells and important to fight against viruses. So I decided to present you some recent work that we have published or that are still in revision. And at the end, I will present you what we are going, what we initiated recently to see how we can find some converging interest. So first, the part on CDC1. So we clearly demonstrated that CDC1 are important for tumor immune surveillance. First, by analyzing a cohort of patients by multiplex immunofluorescence, where we analyze the expression of a receptor that is specific of CDC1. And we demonstrated that the presence of CDC1 in tumor is associated to good prognosis in triple negative breast cancer. And this data in human was confirmed by an experimental work in a preclinical model of TNBC in mice that we did in collaboration with the group of Marc Dallot in Marseille, where we showed that in the CDC1 knockout mice, it means mice that do not have CDC1, the tumor cells will grow and develop, in contrast to the control mice where the tumor is controlled. We went a little bit further and we looked at the function of the CDC1 in the tumor, and in particular, there are many papers showing that these cells are important to cross-present the antigen, and that's why they are important for anti-tumor immunity. But there were no data, especially in human, but also in mouse, showing the role of type 3 interferon produced by these cells in the tumor context. There are many papers in the context of infectious disease, but not in the context of tumors. So we were the first to demonstrate here by in situ hybridization that the CDC1 in green were able to produce type 3 interferon here in red, or in pink, sorry, directly in the tumor microenvironment. And it is illustrated here for many patients. And what was interesting is that when we look at the prognosis impact of the expression of interferon lambda or its receptor in public databases by RNA transcriptomic analysis, we observe that the presence of the expression of these two molecules is associated to good prognosis in breast cancer. So then we wanted to know what kind of cells will respond potentially to the type 3 interferon that is produced by the CDC1 in the tumor microenvironment. And so we initiated like a screen to see which immune cells in human coming from the blood of healthy donors will have the propensity to respond to type 1 or type 3 interferon by phosphorylating STAT1, which is a signaling molecule downstream the two receptors. So as you can see in blue, every immune cell is able to respond to a type 1 interferon by phosphorylating STAT1. But for the type 3 interferon, it was interesting to see that only plasmacytoid DC here in red are able to phosphorylate STAT1 in response to type 3 interferon, suggesting that these cells are the specific responder to type 3 interferon produced by CDC1. So we then illustrate this potential interaction in situ by multiplex immunofluorescence by showing here in the dotted line that there are strong interaction or proximity between the PDC and the CDC1 in green in tumor sections. So all this data led us to propose this kind of model where we observe that CDC1 are activated inside tumors where they produce type 3 interferon, and this type 3 interferon will be important to induce PDC activation. I didn't show you the data for sake of time, but we demonstrated that the type 3 interferon is important to improve the production of type 1 interferon by PDC even in tumors, and that this collaboration between CDC1 and PDC is important to initiate CD8 T cell recruitment that will be activated by CDC1 in the tumor microenvironment. So then another project that we also initiated recently in the lab was to analyze the role of NK cells in immune surveillance, in cancer immune surveillance, especially because you probably all know that NK cells are very important effector for anti-tumor immunity, in particular in the early stage of tumor development, thanks to their two main functions, supportive functions through the production of cytokine and chemokine that will be important to recruit and activate other immune cells, but also their cytotoxic function like NKT cells or like mate cells that were presented before. And this activation of NK cells is controlled by the balance of activating and inhibitory receptors, and in particular, it is not illustrated on the slide, but the cytokines, as previously mentioned also by Agnes and Alexandre, are very important to trigger NK cell activation, especially IL-12, IL-18, and so on. And in this context, we were particularly interested in by analyzing the role of IL-33, which is a member of the IL-1 family, so close to IL-18 that was known to activate NK cells, to analyze the role of this cytokine in NK activation. Why? Because IL-33 is a cytokine that is expressed in the nuclei of the cells, mainly, that is retained in the nuclei, and that will be released upon stresses, and stresses that can be found in the tumor microenvironment. So we hypothesized that this IL-33 could act as an alarmin and activate NK cells in the context of tumor immune surveillance. So we first demonstrated that NK cells spontaneously do not express the receptor of IL-33, that is called ST2, but upon IL-12 activation for 1 day, they will acquire the expression of ST2 and will become responsive to the alarmin IL-33. So we addressed two main questions. The first one was what are the features of these ST2 plus NK cells, and can this population be targeted in tumors? So it's just 2 slides of summary of the main data that are under revision in Science Immunology. Is that this population of NK cells, ST2 positive, is an intermediate between the 2 main conventional NK cell population, the one that is called the CD56 bright and the other one that is CD56 dim. And it is here a transcriptomic, bulk transcriptomic analysis by showing this intermediate phenotype with specific genes to ST2 population, but also common gene with bright or common gene with dim NK cells. And this intermediate phenotype confers a kind of polyfunctionality to NK cells because these cells will be able to secrete cytokines, to be cytotoxic, and to proliferate. So these cells has a strong potential to have anti-tumor function, and that's what we addressed either in the human settings, but also in mouse model after. And so what we observe is that when we got access to patient blood and patient tumors, we investigate the capacity of the NK cells to produce interferon gamma in response to IL-12 and IL-33 activation, and we saw that in both contexts, these tumor-associated NK cells were able to respond, even though with to lower extent for the one coming from tumor, but it's in agreement with what is known in the literature regarding the dysfunction of NK in tumors. And interestingly, when we combine a high score for NK and a high score for IL-33, it has a very good impact on the survival of the patient in breast cancer. And using this mammary tumor model, orthotopic mammary tumor model that is known to be controlled by NK cells, we also demonstrated that the administration of IL-12 and IL-33 has a strong therapeutic effect that prevents tumor development. But when we deplete NK cells, that are the dotted green line, we partially lose the therapeutic effect of the treatment, meaning that NK cells are indeed important to mediate this anti-tumor effect of the combination of IL-12 and IL-33. So this led to this final scheme of this work, of these 2 works, so showing that there is a tripartite interaction, NK cells being important to activate the XCR1, CDC1 through their production of interferon gamma, and that the CDC1 are important to induce this ST2 population through the production of IL-12. These cells, in response to IL-33, will produce interferon gamma that is important to activate CDC1. And here is the presentation, the result of the data on PDC and DC interaction. So it means that this crosstalk between NK, CDC1, and PDC is very important in anti-tumor immune response. And now I would like to share with you some very recent results we obtained with an ongoing project on immune surveillance of preneoplastic stages. So now we decided to investigate the role of these populations, dendritic cells, NK cells, but also recently we also initiate work on neutrophils, but in different stages, which is the preneoplasia, so the stage just before initiating tumor development. So we initiate two kinds of analyses, one that will take into account the spatial structure of the tissue, but also the time with the tumor progression. And our hypothesis is that at the preneoplastic stage here, immune surveillance pathways are efficiently activated in innate immune cells, and in particular in the one we are interested in here. And the objective is to discover a new immune surveillance pathway in this immune cell population that are mainly innate immune cells during tumor initiation that could be used to prevent tumor emergence, to make like cancer interception, and reactivate anti-tumor immunity in advanced tumors where the mechanisms have been inhibited. And the idea is to generate new concepts and new targets for cancer patients or patients with high risk to develop cancer. So to do that, we have developed two different models because it's not so easy to analyze the role of immune cells at preneoplastic stage before the tumor development. So we have human access to human tissue from breast cancer, but also from colorectal cancer. And we have from patients with colorectal tissue, not only cancer, we have also polyp and normal tissue. And we have also developed a spontaneous mammary tumor model that will develop tumors a long time. So this is the model. I will only focus on the mammary tumor model and not on colon project, but we can discuss after if you want. So we use a transgenic mouse model that express the BRCA1 under the control of the beta-lactoglobulin that is targeted specifically in the mammary gland in a P53 heterozygous context. And this tumor, following different cycle of gestation and lactation, will develop tumors. So it's an inducible model that will recapitulate the pathology more than if you implant a tumor cell line in the mice. So we have access to this kind of tissue, healthy tissue, early stages, and also invasive carcinoma. And what we first decided to do is to analyze the presence and the function of the immune cells in this different type of tissue. And we were able to identify some differences at the level of, for example, infiltration of NK cells with a peak at the preneoplastic stage, for example. And we were also able to show that these cells are functional and can produce the typical cytokine for them, like interferon alpha for PDC, interferon type 3 for CDC1, and IL-12 for CDC2. So this model was suitable to analyze the role of the innate immune cells in tumor initiation. So to perform a comprehensive analysis of these changes across tumorigenesis, we decided to perform single-cell RNA-seq analysis. And there was a first challenge that was to have enough cells coming from this innate immune part, because these cells are rare and they are less abundant than T cells or macrophages. So we decided to use a strategy by cell sorting to equilibrate the number of the cells and to have the same number of, for example, of NK cells like CDC1, CDC2, and B cells, T cells, and so on. So it's the first study that uses this kind of approach to be sure to analyze in depth the innate immune cells in the tissue. So we were able to sequence more than 40,000 cells, total cells, and we have started to investigate the bioinformatic analysis of the tumor, the epithelial or tumor cells, but also of the dendritic cells here and the family, and also of NK cells. And the first results show that indeed we have different clusters of NK and the innate lymphoid cells, but also different clusters of DC macrophages, and some seem to be specific to different tissue stages, with some cluster being enriched in normal tissue, some in preneoplastic tissue, and some in tumor tissue. So this validates our hypothesis that the immune system, that the innate immune cells are different between the different stages of tumor development, and then we can identify some specificities at these preneoplastic stages. So this project started recently. What we are doing is trying to analyze the cell states along progression with the single-cell RNA-seq. The idea is to identify pathways that are activated at early stages by integrating the temporal dimension in the project through bioinformatics and the modeling of the data. And we also would like to integrate what we observe in human for the colon part and what we observe in the mice for the breast cancer part. This will be also connected to in situ localization and analysis of interaction of the cells by multiplex IF, but also we plan to do spatial transcriptomics. And the idea is to identify spatial crosstalk between the different cells of interest, and to identify potential therapeutic targets that we will validate in cohort of patients by multiplex IF, but also in vivo by using KO mice. And the idea is to ultimately analyze the biology of this potential target and develop new medicine, new drugs to target this molecule by using CRISPR or MABS approaches. So this is a challenging project, and we have a lot of challenges remaining, and specifically for the different steps of the project, either from the single-cell RNA-seq data to the target identification. In particular, it's a work that needs a lot of bioinformatics, of integration, of modeling, and so we will be very open for collaboration on this part and for help. We also have some issues still regarding the step from target identification to target validation, and in particular regarding the models, where we're starting thinking about organoid models or in vitro model with immunoorganoid that we still need to develop in the lab, but also CRISPR screen approaches that we are not familiar with in the lab. And we also would like to extend the multiplex IF more than 7 colors to have a very comprehensive analysis of the immune microenvironment. And finally, we are also open for collaboration with biotech for the development of drug targeting the molecules we have identified. So I'm done. I just like to thank all the team, all the people who are involved in the team. I will not say one by one, but I would like to thank all of them and all our collaborators, of course, and you for your attention. Thank you. Okay.
I'd like to invite all the speakers so they can have a seat here, and I will open for questions. But before, just because they're just arriving, I'd like to say something that Nathalie started when she introduced the presentation, that when we do science like you do here, we're trying just starting doing some research, and also are in a funding agency like FAPESP, we always have this thought: how can we measure the amount of money that we put in science, that we pay for them to do the research? But here we have just very nice example that Alex was a young investigation. He was PhD postdoc. Rodrigo went to France, supported by FAPESP, of course, the other agencies. But we have like an idea: how can we actually measure that this is worth to send students abroad to make connections? So I think for us, it's very nice. We have to figure it out how you can make this connection. So Before I open, I also want to say, because I'm seeing a lot of students and I like this, if you want to make questions in Portuguese, we can translate and then they can answer in English. So this is a way that actually you feel comfortable to make questions. So don't be shy. So I will open now for the first student. It's very difficult, but okay for everyone who wants to make questions. Okay, so Professor Sergio, then I will have quite a lot of questions.
First of all, I'd like to congratulate all of the speakers, wonderful talks. And my question goes to Agnes. I was wondering if in your model of a COVID, the cytotoxicity induced by MATE is only the mechanism to induce pathology, or could you relate to anything? Because in the leishmaniasis model, CD8 positive T cell expressing granzyme do cytotoxicity, but increase inflammasome activation and inflammation.
Okay, look, of course, I will not say that the CD8 cells didn't play any role, nor the NK cells and other cells. I'm sure, you know, unfortunately, the COVID was such a terrible disease because what we saw, I mean, it was clear in the slides and in other papers, we never seen before in other situations. Okay, I do think the CD8s can really participate to the killing, because, but in our studies, I didn't study the specific CD8 T cells against the virus. But when I do compare with other cell population, it's like all CD8, all CD4. Look, maybe if I have a specific peptide tetramers to redetect a specific CD8 population, it might be as bad as the MATE cells. But what was really clear is this shift from terpinterferon to inflammasome. And we mentioned, and Nathalie mentioned again, the cytokines. L18 receptor is mainly expressed on MATE cells, much more than on NK cells and much more than NKT cells. And in fact, for L12 receptor as well, it's much more in NKT than in NK cells. Look, I think these innate like T cells are really the first one to respond, and they respond very, very strongly.
Okay, so maybe I do one question before the others are starting heating up. Alex, you said that you want to find ways to do some immunotherapy, and you mentioned that you found that PKM2 was a nice pathway. Do you think that we can imagine to combine alpha-gal linked with an agonist of PKM2, so only cells that will actually capture alpha-gal will have a chance to internalize this agonist, and then you boost specifically, or you block, because you have both drugs to modulate PKM2 pathway? This could be like one possibility for the way it's open.
Thank you for your question. Yes, we hope so. But what I was thinking about was first boost NKT cells, then make the infusion. Because if you look at the treatment for a tumor, some proposals are to use inhibitors of PKM2. So the problem is when we say inhibitors, we mean translocation. So that's the question. You think that we can boost the glycolytic pathway? I hope so, but I'm still afraid because I don't know how NKT cells will respond to the PKM2. That's my first question that I want to really to answer. I hope in a few years. But thank you for the question.
More questions? I think Zeka here.
First of all, very nice section in immunology. Congratulations also, Niels, to organize these different topics. My question first is for Agnes about the CD39 expression in mite cells. Because this is very interesting in terms of modulation of the cells, but sometimes the expression of CD39 is also associated with exhaustion. So my question is, do you know exactly in which condition these cells start to express CD39, and if there is any cytokine that's known to drive this expression?
Okay, look, in fact, I just showed you the last cartoon, not all the data. But interestingly, we analyzed the MATE cells at different times. Look, for us, the disease you saw arrive like 10, 11 days after the infection with the virus. But we know that in diabetes, often what is key is the initial immunoregulation. Look, if we look at day 3, we have this major increase of CD39. But if we go further and we look like the 8, the 9, they still have this very high expression. And the number of cells, in fact, it's interesting. I don't think they're exhausted. They're really regulatory. And in fact, if we don't block them, they are like acutely activated, and then they lose the numbers. Like in fact, when we treat with acetyl CSP, we have more MAIT cells. Because, you know, like the innate cells, when they get very highly activated, then they die. I mean, it's supposedly what they should do, because if not, we have inflammation. Look, I don't think it's linked to exhaustion. Or, I mean, they might express PD-1, but in other words, I don't think they die. I think they really survive and the regulation.
Perfect. May I, because I would have to have one for each one. Is it possible? Because it's so...
Of course, man.
Yeah, but for Rodrigo, it's very interesting data, actually. It's a lot of data, and I couldn't follow everything. But perhaps I missed something, but your marker is FOLR2. Is there a ligand for this receptor? And what's the really, because you just mentioned about a marker, you know, a marker. But I would like to know if this is really a receptor that has a function and can drive something.
Okay, so my question goes exactly the same. So just to share that we have the same thought, because I think oncology like a lot of this mark. This is FOLR2. Immunology is all about function. So what about folate do in this macrophage? I think it's the same thing that Zeka is asking there.
Yeah. So this question I answered like, I don't know, 100 times already, because everybody wants to know. So we don't know. Actually, because there are 4 receptors for folate. Beta receptor are mostly expressed on myeloid cells, immune cells. Alpha could be expressed by tumor cells and other type of cells. And the gamma and the delta is not so clear. So both alpha and beta could be targeted by folate acid, for example. And there is then I discovered during writing the paper, there is a group in the United States in Purdue University. They developed some kind of approaches to deliver folate acid to those macrophage-like cells. And we really don't know what the folate does in this kind of cells. We found as a marker because it was like a tag to take a look on those macrophages. But we don't know exactly what is happening if you put some concentrated. And then if you come back to the papers in the early days when people start to give folate acid to the pregnant woman, for example, so the placenta is full of those macrophages expressing folate receptor. And so there's probably some kind of regulation of function of those cells when you give more and less. And then our article revealed that folate2 for luminal breast cancer associated to a good prognosis, but there are many papers saying the contrary. Okay. And so it's still, it's all new.
So,
yeah.
And just to finalize, sorry if I'm manipulating for Nathalie, it's a very nice talk with different connections of different cells releasing different cytokines. My question is about the release of the type 3 interferon, the lambda. That's very interesting, and we don't know too much about this. There are some papers showing that STING activation is able to regulate the expression of type 3 interferon. And now we know that there are many clinical trials using STING ligands for cancer. So do you have any connection of STING in your context?
Yes, this is a very important point also. So we didn't investigate what kind of signals in the tumor will trigger type 3 interferon by CDC1, but STING is one candidate with the STING agonist, especially because it has been described that some STING ligands are released during tumor death or tumor stress, and this ligand can enter into DC and can reach the STING in the cytosol, the STING pathway. So that's something we wanted to address. We initiated to activate CDC1 in vitro with STING agonist, and indeed we observed the production of type 3 interferon in response to STING agonist in CDC1, but it was only in vitro with healthy donor cells, and we didn't translate to the tumor context. So I cannot answer, but it's one of the most candidate we have in mind.
And the last one to Alex. In terms of PKM2, so we know that PKM2 has a non-canonical function, so translocating to the nucleus and acting as a co-transcription factor. So do you think that the important role of PKM2 in your context, in your INKT, is to regulate the expression of genes associated with metabolism, or something else that's important for the function of the NKT cells, like interferon? I don't know.
That's a great question. In the work that we published, we show that glycolysis is important. But I'm wondering if in the cytotoxic pathway of NKT cells, maybe transcription upgrading the gluten, HIF1 alpha, all this. I'm not sure yet. I think so. That's the first question that we have to ask. Which is the pathway that we're dealing with? But thank you for your question.
Thank you, Zeka. Okay, I still have two questions, one for Agnes, one for Nathalie. So Agnes, you start also your presentation saying that Alex helped you because he talked about NKT. NKT seems to be a sister of mate cells. So, and you also said a lot about the unique ligand that we have so far is a microbial derivative ligand. Do you think that in the near future, I will come up with some subsets of mate cells in terms of Th17, regulatory, something like this? And also, in this sense, what you saw in COVID data that Sergio asked is about the mucosa in the lungs, in the sense that there was a specific effector phenotype, activation phenotype on a specific subset of cells, or these cells looks like the same as the intestinal mucosa?
I guess it's a complete question. Okay, now regarding mate cells in the mice, they are very different. It's like the mate1, mate17, a bit like the NKT. They are very split. They express either T-beta or gamma T. They are different. But in the human, it's very different. They all express both. And of course, there's many studies ongoing now in epigenetic to understand why they have gamma T, they don't express L17, or why they have T-beta, they don't express interferon gamma. Look, it's clear that what we see in the context of inflammation and very activated mate cells, they tends to express both. A little bit like some CD4 cells when they get like BJ crucial work, when they're very pathogenic, in fact, they express both 17 and gamma. Look, we have the same thing for the mate cells in the patients. Regarding the lung, now we did single cell analysis in the blood, but we can really see the different subsets. And it's very nice because, in fact, even in the human, when you go to young kids, we can find subsets, which nobody have described that before in the adult, but in the kids, we might find some mate1 and mate17 like the mouse model. And we didn't do the single cell in the lung. Look, some people have done it, but with like the total populations, but they have very few mate cells. Look, not enough to survive. But many open questions. And, we have to go further.
This is good. And Nathalie, just to finish, and then we go to the coffee break. I love your presentation, and I feel wonderful to meet you because Rodrigo always talk about how he was in Lyon, and so far all these nights abroad, he went to shows and something like this, never in the lab, and you will always call him back, please do some research. My question is, if I go through your presentation in the beginning, when you start about anti-PD-L1 and all this resistance to the chemotherapy, this new immunotherapy, and then can you combine both? I mean, do you think that what you're saying about subtype of neutrophils that you are saying, this will explain why those patients, they actually don't respond successfully to this immunotherapy and so on?
Yeah, so there are some biomarkers that have been identified for the resistance to immunotherapy, and in particular some type of it's better, it's the way around. You need CDC1, you need a subpopulation of neutrophils to respond to immunotherapy. So probably that the innate, that the way I thinking is that the innate immune system is important to trigger response to anti-PD-1. In particular, you need CDC1 for the antigen presentation. You need one subtype of neutrophils. I don't know exactly why this subtype, but we are also investigating the heterogeneity of neutrophils in our mammary tumor model, but also in the colorectal cancer with the different stages of tumor development. And so probably that we will have a new answer about the role of these cells and how they can help or counteract the anti-PD-1 activity. And our idea is that if we know more about these innate immune cells, we can provide new treatment, new targets that could be combined with anti-PD-1 to enhance the response to the patients.
Fantastic. Any more questions? Oh, yes, Thiago. Can you?
Thank you.
Is this something related to virus infection or something related to endogenous retrovirus elements produced within the tumor microenvironment of breast cancer?
That is a very important point. No, you're totally right. We have a project in the lab dedicated to retrovirus antigen. Retrovirus, endogenous retrovirus, but the project is more related to their role as neoantigen and as target for vaccination, for example. But I am thinking of developing a new project that could be the role of this reactivation of retroviruses, endogenous retroviruses, in the activation of dendritic cells, because it has been shown in other kind of tumors that when they are re-expressed following some kind of treatment, they lead to the expression of interferon pathway, in particular type 1 interferon, and also type 3. So maybe that it can play a role also in the breast tumor. Even, and it's how to say that? We identified the ERVs mostly in the triple negative breast cancer. They are overexpressed in this kind of breast cancer. So that the breast cancer, we investigated the role of CDC1 also. So probably that there is a converging role.
Thank you. Can I ask one more question or not?
Yeah, during the coffee maybe.
Okay, thank you.
So thank you very much. So big applause to them. We go to the coffee break where you can meet them, and then we come back in half an hour for the second immunology session on oncology. Okay.
My name is Leandro Colli. I'm a professor of oncology at Ribeirão Preto Medical School. I will be moderating the section that we're going to talk on oncology. Niels will be jealous because this is actually the best topic. And we're going to start it. We have 4 amazing talks this afternoon. Last but not least, we're going to start with Dr. Marion Alcantara. She's from Institut Curie. She's MD, PhD, and hematologist by training. She's the medical director for Cell Action. She's the president of French Society of Cancer Immunotherapy, and she's a brilliant expert in cell therapy, gonna give us a very good talk today. Thank you very much one more time for being here.
Thank you very much. Thank you. So yes, indeed, I'm hematologist at Institut Curie, and I'm the medical director of a new R&D lab that we have just created at Institut Curie that is called Cell Action, for Cell Therapy Acceleration and Innovation. And I'm going to focus on only one project of Cell Action, so it will be a talk that is going to be different from what you've heard before in the last session. And I'm going to talk about how to improve the efficacy of CAR T cells for primary central nervous system lymphoma that I'm going to talk PCNSL. So what is PCNSL? It's a B-cell derived cancer. It has been now classified as a large B-cell lymphoma of immune privileged sites. Anyway, it's a cancer that arises from a B-cell, but it's exclusively localized in the CNS, mostly the brain, sometimes the meninges, and less often the eye or the spinal cord. The pathophysiology of this disease is not fully understood. It has been hypothesized that the clonal BCR of the clonal B cell could recognize some CNS proteins, but the papers are not really convincing. It's a rare disease representing 300 new cases per year in France, and the median age is 67 years old. It is associated with a poor prognosis, poorer than what we observed with the systemic B-cell, large B-cell lymphomas. In terms of symptoms at diagnosis, most of the patients present with cognitive or behavioral disorders, and believe me, they are often totally crazy, which is very hard to manage for the medical and paramedical team, but also for the family, it's very hard. Also, most of the time, they have gait disorders or motor and sensory deficit, but not really headache. Headache is not a way to enter into this disease. So it's very different from what we observed with classical systemic lymphoma, for which patients are going to come to the doctor because of an adenopathy or any tumor or mass. In France, we have the chance to be very well organized for taking in charge these patients, because we have a French national network that is called the LOC network, and it has been funded by the One Cancer Plan for Rare Cancer. They have a huge national database. Just for you to know, we now have 3,000 patients that are prospectively recorded within this database. So it's really nice data to do very good real-world evidence studies. And the LOC may make recommendations for the treatment of these patients. I think all the centers in France which are taking in charge these patients now do it following the LOC recommendations. And of course, they promote clinical trials and translational research. One of the paper, I took some survival curves of one of the LOC papers. So they analyzed 1,000 newly diagnosed patients with PCNSL. Among these 1,000 patients, around 500 presented with a relapsed or refractory disease. And you can see that for patients who will receive conventional chemotherapy at relapse, the survival is really poor, 22% at 2 years. Patients who are able to receive autologous stem cell transplants do better. We know that this kind of strategy can be curative for these patients, but these stem cell transplants are associated with a high morbidity and very high levels of toxicity. So there are many therapeutic challenges for these patients, mostly due to, the first thing is that the tumor cells are often associated with molecular subgroups of poor prognosis. They are also heterogeneous. This cancer arises in a immunosuppressive microenvironment, and of course you may know that there is this question of blood-brain barrier that can decrease treatment accessibility in the CNS. And on the other side, there is a question of neurotoxicity of the treatments. For a long time, radiotherapy was part of the standard treatment, and we're doing this less and less because now we know that radiotherapy is associated with irreversible cognitive disorders. It's less clear for chemotherapy and immunotherapy. Regarding cell and gene therapies, I think you also know that there are the latest revolution in immuno-oncology. And among cell and gene therapies, there are plenty of things. For example, T-cell infiltrating, tumor infiltrating lymphocytes. And the CAR T-cells have made a huge breakthrough in the treatment of many hematological malignancies. So I'm going to talk a bit more about CAR T-cells, of course, but just for you to know, the first patient treated with second-generation CD19 CAR T-cell was not Emily Whitehead, but she was one of the first. Emily Whitehead was a young patient. I think she was 10 years old. She was treated 11 years. Yes, she was treated 11 years ago. And she was cured. She had a B-cell acute lymphoblastic leukemia with no therapeutic options. She has been cured with one infusion of anti-CD19 CAR T-cells. And since then, you can observe here, or maybe it's a bit small, but anyway, there is a growing number of indications and approvals for CD19 CAR T-cells or BCMA CAR T-cells for many hematological malignancies. And also we are using now these CAR T-cells earlier and earlier in the different lines of treatments. So what are CAR T-cells? What are we talking about here? There are T-cells that are genetically modified either by a retro or lentivirus. There are plenty of other ways to do it, but mostly LVVs. And they are genetically modified to express at the cell surface SCFV, so a structure that is going to recognize an antigen that is not presented through the MHC complex, and it is fused with the TCR activation domains in the intracellular part. We always think that CAR T-cells are a very new story and very American one, but it's not true. Actually, the first generation of CAR T-cells were published in the 90s by an Israeli guy, but it didn't work correctly because there was no in vivo expansion. Then the second generation of CAR T-cells, the ones that are currently commercialized for the treatment of lymphomas or myeloma, have been developed by a French guy who is called Michel Sadelain, but actually is more American than French now because I think he's at MSK-CC for more than 30 years. Anyway, and also we have to remind that Carl June at UPenn, who is a true American, did the same. So Carl June and Michel Sadelain both developed second-generation CAR T-cells by adding a co-stimulatory domain in the construct. Carl June had added the 4-1BB domain, and Michel Sadelain added the CD28. I'm making a long story short, of course. And there are different third and fourth generation maybe coming in the future. In terms of a clinical journey for the patient, so when we talk about autologous CAR T-cells, the patient first have to undergo leukapheresis. The T-cells are then sent to the cell therapy manufacturing. The T-cells are isolated, they are activated and transduced. And then we wait for the expansion of the CAR T-cells. And after, we infuse these modified T-cells to the patients, but before the patients had to undergo lymphodepletive chemotherapy. And we now know that this is very important for even for the autologous CAR T-cells to be active. So if we talk about large B-cell lymphomas, systemic large B-cell lymphomas, there are now different CD19 CAR T-cells that are commercially approved. Axi-cell, Tisa-cell, and Lyso-cell. I'm not going to enter into the details, but most of them, notably Axi-cell and Lyso-cell, have shown very, very nice results in third line of treatment and second line of therapy now. They all use the same anti-CD19 binder that is called FMC63, and then there are some differences in the costim domain that is used here, also in the type of gene transfer, retrovirus for axicel and LVVs for the others too. You may also know that CAR T cells are associated with a specific toxicity. So the first one is cytokine release syndrome. It is really linked to the fact that when a CAR T cell encounters the cognate antigen, it will of course activate. This activation will lead to the secretion of different cytokines such as interferon gamma, GM-CSF, TNF also, that will in turn activate innate immune cells. And the most important here are macrophages. There is also an indirect interaction through the CD40 ligand in between the CAR T cells and the macrophages. Macrophages are going to secrete different cytokines such as IL-6 and IL-1, and these major cytokines are associated to the endothelial damages, leading to vascular leakness. And so first it always starts with a fever, but then you have this endothelial damage that leads to hypotension, eventually capillary leak syndrome, respiratory failure, and some patients unfortunately die from multi-organ failure here. Also, the other specific toxicity with anti-CD19 CAR T cells is this immune effector cell-associated neurotoxicity syndrome, or ICANS. In terms of clinical presentation, it goes from writing impairment up to cerebral edema, and there are also, unfortunately, some patients who died from grade 5 ICANS. The pathophysiology of ICANS is well less understood, but everything is again linked to the increased vascular permeability and the cytokine storm. There are also unpublished data showing that microglial activation is also related to these ICANS. One important thing when we think about PCNSL is this paper that has been published a few years ago. In these papers, the authors showed that using single-cell RNA-seq, they were able to see that mural cells, which are cells that surround the endothelium in the CNS, they express CD19 in human, but not in mice. And with this, they hypothesized that neurotoxicity could be an on-target off-tumor effect of CD19 CAR T cells. So there were major concerns about neurotoxicity of anti-CD19 CAR T cells when we were going to use it for PCNSL patients, because the tumor is within the CNS, sorry. So most of the patients with PCNSLs were excluded from the pivotal trials with axicel, tisacel, lysocel, and so on. Anyway, for different reasons, some teams did treat patients with anti-CD19 CAR T cells, and we did it within the French LOC network. So we published 2 papers. The first analysis was done in 2021, and the latest one has been very recently published. So from March 2020 to November 2022, we treated 25 patients with tisacel or axicel. The median age was 68 years old, and they were all heavily pretreated patients. I'm not going again to enter into the details, but of course we can talk about it after. You can observe here a very nice complete remission with a patient who presented with a big right frontal lesion at baseline, and she is still in complete response some years after the CAR T infusion. So we observed a very nice rate of overall of complete response, 64%, 20% partial response, and interestingly, the responses were observed in each of the CNS compartments, so the brain, the CSF, and the eye. Unless, of course, it's very difficult to assess the response in the eye, so we have fewer patients here. So we know that there are high response rates that seems similar to what is reported for systemic lymphomas. It has been published that the tolerance profile is similar too. Here, it's not really sure, and in France, we have an ongoing study regarding the risk of prolonged neurotoxicity. So here it's important to know that in terms of percentage, we have the same percentage of ICANS for PCNSL patients as compared to systemic lymphoma patients. But we saw in our study a first signal because 3 patients among the 25 infused here presented with grade 4 ICANS lasting more than 6 months, which is totally unexpected with this toxicity for systemic lymphomas. You can observe here the overall survival of the patients. So yes, the patients do respond very high, but they still relapse and die. And we do not really see this very nice plateau at 40% that we observe with systemic lymphoma and anti-CD19 CAR T cells. Anyway, there is no commercial authorization for these patients, and we need to improve long-term efficacy. I'm showing you here 2 slides that were presented very recently at ASCO by a colleague in Dana-Farber. They have an ongoing clinical trial focusing on axi-cel for PCNSL patients. They have treated so far 18 patients, among them 13 PCNSL patients. I'm not showing you the results in terms of efficacy because it's really similar to what we observed and we published, but they did very interesting ancillary studies with different CSF and blood samples in this clinical trial. And there are 2 things here. The first thing they observed is that CD8 CSF, as compared to CD8 CAR T cells in the blood, they express more exhaustion-associated genes. So you could say, okay, but that's normal because the tumor is within the CSF, so the CAR T cells encounter more the tumor, so they do express more exhaustion markers. But more interesting, they were able to show here that when they did this differential expression analysis in between the patients, so for CD8 CAR T cells in the CSF, but comparing patients who obtained a complete remission as opposed to those who had progressive disease, they also observed a high upregulation of BLIMP1, also associated with exhaustion. So maybe it's really this question of exhaustion is very important for CAR T in the CNS, also because we know that there is a highly immunosuppressive microenvironment in the CNS with astrocytes and microglia cells, etc. So we went back to the lab and we are currently actively working on how to improve CAR T cells for PCNSL. We have 2, and we are doing both the modification of the CAR design and epigenetic reprogramming of the CAR T cells. So we have a nice collaboration with Michel Sadelin, and this is why we wanted to take his very nice backbone of 1XX CAR T. So what Michel published a few years ago, in his lab, they hypothesized that the redundancy of CD28 and the full CD3 zeta signaling, when you have the complete 3 item domains, might foster counterproductive T cell differentiation and exhaustion. So they induced mutations in all or each of the 3 items domain of the CD3 zeta, and they were able to show that the 1XX here, so the one that has a mutation, of course non-functional mutation, in the 2 distal items of the CD3 zeta, they really outperform the other constructs in a core stress in vivo with an in vivo model of B-cell acute lymphoblastic leukemia. There are plenty of mechanistic analysis in the paper. To make, again, a very long story short, they showed that when doing these 1XX mutations, they have a better balance of effector and memory programs. And then they went to a phase 1 clinical trial very quickly, as MSK-CC knows. And they showed this at the last ASH meeting. So they've treated 28 patients with aggressive B-cell lymphoma. The first very important thing, maybe the most important, is that the recommended phase 2 dose here is 25 million. Just for you to know, Lysocell that is currently commercialized, it's 100 million total dose. So really, when using this kind of construct, you're able to decrease the recommended phase 2 dose. And this is also related to a better toxicity profile because they only observed 4% of more than grade 3 CRS and 7% of more than grade 3 ICANS. And still they showed very nice response rates with an overall response rate of more than 80%. So we want to use this 1XX design in terms of really the CAR. Then how to improve again the 1XX? We have been working on epigenetic reprogramming of the CAR T cells. So SUV39H1 that I'm going to call SUV for the last of the talk. It's an histone 3-methyltransferase, which leads to a chromatin compaction and gene silencing. Sebastian Amigorena, who I worked a lot with, has worked for many, many years, more than 10 years, on the role of epigenetic reprogramming of T-cell fate. And he published different nice papers. The first one showing that, it's all in mice here, showing that in naive CD4 T cells, SUV39H1 inhibits Th1 loci, thus ensuring a Th2 stability. Then when considering naive CD8 T cells, SUV will inhibit the expression of stemness memory genes, leading also to this CD8 effector phenotype. And very interestingly, the inhibition of SUV make able a reprogramming with anti-PD-1 to a memory-like exhausted CD8 T cells, meaning that these T cells here do express exhaustion markers, but still they are able to kill the tumor, and they also have a memory program. So everything here was done in mice. The question, of course, naturally was what could be the role of inhibiting SUV in human CAR T cells? And again, I'm making two very long story short. We published back-to-back with Michel Sadelin very recently in 2024 the efficacy of inhibiting SUV in CAR T cells. So what is very interesting here is that we did it back-to-back in different models with different CAR designs. We used a 41BB second-generation CAR in a solid tumor model. Michel used a CD28 second-generation CAR in a leukemia model. And I'm focusing, of course, on our work and not his. So we set up a solid tumor model by injecting IV the A549 cell line, which is an adenocarcinoma cell line. And when you inject it IV in NSG mice, it goes directly to the lung. This cell line has been genetically modified to express CD19. What you can appreciate here is that when the mice are treated with the low doses of CAR T cells, you can see that at the beginning, the control CAR T or the SUV knockout CAR T do the same, but around day 60, all the mice treated with the control CAR T cells finally relapse and die, which is not the case of the mice treated with SUV knockout CAR T cells. And when using higher doses of CAR T cells, so all the mice are virtually cured, either with the control or the SUV knockout CAR T cells. But when we re-challenge the mice by re-injecting tumor cells without putting new CAR T cells here, you can see that the mice treated with the control CAR are not able to control the re-challenge, whereas it's not the case for the mice treated with the SUV knockout CAR T cells. So we performed different single-cell RNA-seq and single-cell attack-seq studies, and the summary is here, meaning that when you inactivate SUV39H1 in CAR T cells, you still have the stem memory genes that are expressed and functional together with the effector gene programs. So you increase the functional persistence of the CAR T cells. Of course, we wondered how this was going to work in a PCNSL model. So we developed a PCNSL model using this cell line that is derived from a patient who died with a PCNSL in Japan in the 80s. It expresses highly CD19, and it has a phenotype of non-GC DLBCL, which is very classical in PCNSL patients. High levels of IL-10. It secretes high levels of IL-10, which is also what we observed in patients. And there is a downregulation of HLA-1, which is also something that we often observe in the tumor samples from patients. We set up an in vitro spheroid model because it's, well, we think that 3D models are really better to analyze the in vitro efficacy of CAR T cells as compared to 2D models. And again, we can discuss this after. I hope it's going to work, but it's not because I have the pointer. So you were supposed to see a nice killing of the CAR T cells. Yes, you're going to see it. A nice killing of the TKGFP cells by the CAR T cells here. And also we set up an in vivo model by injecting into the striatum of the mice using this stereotaxic device. We inject TK expressing luciferase here. Sorry. Yes, it's okay. We also observed that TK cells induce a strong astrogliosis in blue and microglial activation in red. So it really mimics as best as we can what we observe in patients. And when we treated, of course, so we wanted to treat the mice here with CAR T cells. At the beginning, we were working on classical second generation 4-1BB CAR T cells. And as you can observe here, we showed an increased survival of the mice treated with SUV knockout CAR T cells. It's not statistically significant here because we had few mice per group because it's a tricky model, so you cannot have 60 mice per group here, and we wanted to test different doses. We moved to the 1XX backbone, doing the SUV knockout plus 1XX. We wanted to analyze the phenotypic differences between SUV knockout and control CAR T cells. So here we did an ex vivo analysis by sacrificing the mice at day 25, and we performed a multicolor, 30-color spectral flow cytometry analysis here. And here are some examples of what we observed, that when we silence SUV in these 1XX CAR T cells, we increased the proportion of cells expressing memory markers, such as CD127 here in CD4 CAR T cells, and we reduced the dysfunctional exhausted phenotype, sorry, as the expression here of PD1 CD39 in CD8 CAR T cells. Again, there are few mice per group, and we're repeating this. So all this in an in vivo model of PCNSL. So the goal here is to launch a phase 1 clinical trial. I forgot to tell you that there is a spin-off that has the patent for this inhibition of SUV. It's a spin-off of both Institut Curie and MSK-ACC. So patent of SUV inhibition in immune cells. So the plan here is to do this phase 1/2 clinical trial, including relapsed refractory PCNSL patients that have received more than one prior therapy and who would be not eligible for autologous stem cell transplant. It will be a very conventional trial for CAR T cells. Patients are enrolled, then they will have leukapheresis, CAR T manufacturing, lymphodepletion conditioning, the CD19 1XX SUV knockout CAR T infusion, and then of course will be closely monitored for toxicity and efficacy together with nice ancillary studies. And this is why I came here, and you all know that I was in Ribeirão Preto at the beginning of the week, and I came here because we aim at doing this phase 1 clinical trial in collaboration with our colleagues in hematology at Ribeirão Preto. The goal would be to include patients both here in Brazil and in France, 10 here, 15 in France, and to have 2 sites for the CAR T production, the Nutera Hemocentro, I think it's correct because I'm still a bit lost with everything, and one GMP manufacturing facility in France. So now we are doing the process development. I'm not going to enter too much into the details, but we are currently working on this to set up a nice process and a robust process using the Clinimax Prodigy, which is an automated closed system, together with an electroporator, because for the knockout we used CRISPR-Cas9 and we need electroporation of the cells. We also plan to include in the construction the EGFRT. This is important because as we will do gene editing, working on playing on epigenetic reprogramming. So of course we never know, even if nothing happened in mice, it doesn't mean nothing will happen in patients. So we never know this. Using this EGFRT will make us able to infuse cetuximab in case of toxicity, and cetuximab will destroy the cells expressing EGFRT. So the plan is this: autologous T-cell isolation from leukapheresis, LVV transduction to introduce the 1XX score and the EGFR-T. Here we will have a step of selection based on EGFR expression, the SUV knockout via CRISPR-Cas9, expansion, or Western cryopreservation. And our goal is to get a GMP-ready CD19 1XX SUV knockout CAR-T for the first trimester of 2025. So this is the conclusion. I showed you that first we started with retrospective studies for PCNSL patients treated with commercial CAR T cells. We showed that it could be improved, go back to the bench, working on epigenetic reprogramming. And with this, we aim to launch future clinical trials together with ancillary studies, because again, it's very important to understand why it works or not in human patients. So I will finish with acknowledging, of course, all the team at Selection with a nice collaboration we have with U932. And I must admit that we have plenty of students coming from South America, but not too much from Brazil, as Rodrigo knows. Thank you very much. What is this? Okay.
Thank you very much, Marion. It was an amazing presentation. We're going to move to the next speaker, so we're going to have all the questions at the end of the section. So it's my pleasure to invite Professor Miriam Galvonas, Associate Professor at UNIFESP. She did her PhD in Ludwig, and she's an expert in molecular oncology. She's going to give us, it's opening, talking about the epigenetics alteration and oxidative stress. Thank you very much for taking our invitation.
Pointers aqui. Good afternoon. First, I'd like to thank FAPESP and Dr. Leandro Colli for the kind invitation. And different for this nice presentation in clinical oncology, mine will be in basic oncology. So I will present here 2 experimental models of melanoma established in my lab for the study of tumor progression, phenotypic plasticity, and tumor dormancy. So melanomas are tumors extremely aggressive. They are the most deadly form of skin cancer and prone to metastasis. And they are also drug resistant, even with the significant advances in modalities, therapeutic modalities such as target therapies and immunotherapies. More than 0.5% of tumors of patients become non-responsive. One factor contributing to this drug resistance is the phenotypic plasticity presented by melanoma cells. And as other tumors, melanomas can remain in a dormant state for months or years and then relapse. So the molecular alterations involved in tumor progression and controlling the phenotypic plasticity and tumor dormancy, understanding these molecular alterations are essential for the identification of drug targets and also to establish more efficient therapies. And for this, we need experimental models. So here I will present 2 experimental models. One is a in vitro cellular model for studying tumor progression and phenotypic plasticity, and the other is an in vivo model of tumor dormancy. So beginning with the in vitro model, We know that melanomas are tumors arising from the malignant transformation of melanocytes. In normal human skin, melanocytes rest on the basal layer of epidermis, where they interact with a great number of keratinocytes and also with the extracellular matrix components of the basement membrane. These interactions are fundamental to the correct function of melanocytes. So the loss of these interactions may contribute to melanoma, malignant transformation and melanoma progression. So based on this, we established this in vitro model. So we established different cell lines after subjecting normal non-tumorigenic melanocytes named melanae, they are murine melanocytes, to sequential cycles of deadhesion and readhesion. So by doing this, we established pre-malignant melanocytes, an example is 4C. We established tumorigenic but non-metastatic melanoma cells, an example is 4C11 minus. And we established metastatic melanoma cells as 4C11 plus. This is a linear model of malignant transformation and melanoma progression, in which one cell line was derived from the other one. So this model is now well characterized. Here you can see that melanocytes and metastatic cells present a differentiated morphology. They are melanocytic, different from the intermediate cell lines, that they are amelanotic and undifferentiated and mesenchymal, they present a mesenchymal-like phenotype. And as I said, this one is metastatic. So this figure shows main signaling pathways altered in human melanoma. And the red circles show alterations found in our murine model. So the main signaling pathways altered in human melanoma are also altered in our murine model, indicating that this model recapitulates what is seen in human melanoma. So trying to understand to identify the molecular alterations related in early and late stages of this progression, we performed some analysis, including the transcriptome, methylome, microRNA expression profile, hydroxymethylome, and the global level of histone modifications. These alterations were classified in 3 different signatures. The malignancy signature, in which the alteration occurs from the beginning to the end of the progression, a plasticity signature, in which the undifferentiated and mesenchymal-like cells, the alteration is present on the cells different from the differentiated cells, and a metastatic signature, in which the alteration is present only in the metastatic cell lines. All of these alterations are always, when possible, validated using data from human melanoma cohorts. So these alterations are studied in terms of their role in melanoma biology and their potential use as drug targets or prognostic biomarkers. Here I brought only one example. TIMP1. TIMP1 is a molecule that has a known role, a known tumor suppressive role as a inhibitor of metalloproteinase, but this molecule can present also oncogenic roles. In this model, we observed an increased expression of TIMP1, and this expression is a result of TIMP1 promoter demethylation. What we've done was to overexpress TIMP1 in melanocytes that do not express TIMP1. And by doing this, melanocytes become able to form colonies in soft agar, a hallmark of malignant transformation. And by overexpressing TIMP1 in metastatic cells that already express TIMP1, the cells become able to form much more colonies in soft agar and also much more metastatic colonies in the lung in vivo. And that TIMP1 expression can be found increased in melanoma compared to benign nevi and normal skin in human. Regarding drug targets, by analyzing all histone modifications by mass spec, we found in the malignancy signature decreased levels of the acetylation of lysine 5 and lysine 8 of histone 4. In the plasticity signature, we found increased levels of D and trimethylation of lysine 20 of histone 3. And in the metastatic signature, we found decreased levels of demethylation of lysine 36 of histone 3.3. So what we did was to evaluate the effect of inhibitors of the enzymes responsible for these modifications. Here we have an example. Entinostat is HDAC inhibitor. We observed that by combining entinostat with a MEK inhibitor, delays the tumor growth in vivo. And the other inhibitors sensitize metastatic melanoma cells to ANOX. Regarding prognostic biomarkers, by analyzing the transcriptome data according to these 3 different signatures, we found hundreds of transcripts in which their expression correlates with melanoma-specific survival in 2 independent melanoma cohorts, human cohorts, TCGA and Leeds melanoma cohort. By analyzing the methylome data, we also found sets of CPGs, both in the malignancy and metastasis signature, in which the methylation status correlates with poor survival. So, as I said, the this in vivo model of melanoma progression is also a model for studying phenotypic plasticity. So we know that melanomas are tumors extremely heterogeneous, presenting diverse cell subpopulations with different transcriptional signatures. And we know that melanoma cells dynamically change their phenotype during metastatic dissemination and also after therapy exposure. The 2 main phenotypes presented by melanoma cells were first described by Hoek in 2006. They are a proliferative phenotype in which the cells are pigmented, highly proliferative, express high levels of MTF and low levels of stimulus factors. And the other phenotype is an invasive phenotype in which the cells present an undifferentiated phenotype, a melanotic, low proliferative, with low levels of MTF and high levels of stimulus factors. Later, other researchers described intermediate states, differentiated states in melanoma cells. So what we did was to compare the transcriptome of our murine melanoma cells with the transcriptome of human melanoma cells presenting different differentiation states. And what we found is that the premalignant melanocytes and non-metastatic melanoma cells corresponds to undifferentiated and undifferentiated neurocrest cell, like corresponded to in human. And our metastatic melanoma cell corresponds to transitory and melanocytic phenotypes. So our murine model follows the multi-stage differentiation model for human melanoma. Now we are trying to identify possible regulators of this phenotypic switch. We have some candidates, such as the long no-coding RNA DLX4OS, and also some microRNAs, such as microRNA 322.5p in human 424, microRNA 125A, and microRNA 138. So maybe they can be possible therapeutic targets. And finally, the in vivo model of tumor dormancy. We know that non-metastatic tumors, when treated, can regret, can be cured, or can remain in a dormant state, as I said, for months or years. And then these tumors can awake, can relapse. So we identified a number of melanoma cells, metastatic melanoma cells, in which when the cells is injected in the subcutaneous tissue in the mice, in immunocompetent mice, these cells do not form tumors. But these cells form tumors in immunodeficient mice, indicating an immunomediated tumor dormancy state. So what we also observed is that if we co-inject with the subtumorigenic dose of melanoma cells a great number of apoptotic cells, these apoptotic cells can be melanocytes, fibroblasts, or even irradiated melanoma cells, all mice present tumors, fast-growing tumors. So this, when we analyze the tissue after 6 hours, 24 hours, 72 hours, we observe an intense inflammatory infiltrate. Then we asked if we co-inject with the subtumorigenic dose of melanoma cells inflammatory agent, such as LPS. And the same is observed. LPS is able to awake this small subtumorigenic dose of melanoma cells. And interestingly, treatments such as radiotherapy and chemotherapy induces apoptosis, massive apoptosis. So maybe these conditions, if a small number of melanoma cells remain after this treatment, this can be favored by the environment containing an acute inflammation. So now what we observed is that if we treat mice with a demethylating agent before the co-injection, there is no inhibition. But if we treat animals after the co-injection, there is a significant inhibition. So indicating that there is an epigenetic regulation of the interplay between tumor cells and immune cells. And now we are analyzing tumors coming from non-treated mice and treated mice treated with a demethylating agent. Regarding to the transcriptome, the methylome, the immune infiltrate, we are characterizing the immune infiltrate, and I hope we can perform in the spatial transcriptomics, because our university got a grant for a multi-user equipment for spatial transcriptomics. And I hope we can do this soon. So in summary, the cellular model of tumor progression and phenotypic plasticity, as I said, is a linear model of tumor progression in which cell lines present the same genetic background. This model is suitable for studying molecular alterations involved in early and late stages of the progression. This model is a murine model, but recapitulate what is seen in human melanoma, and also recapitulate the phenotypic states observed in human melanoma. And importantly, both models are immunocompetent models, so we can analyze different aspects of tumors in an immunocompetent environment. So I would like to thank all students involved in this work, all collaborators, hoping to have a French collaborator. And I would like to thank the funding agency, especially FAPESP. And it's this. Thank you.
Thank you, Professor Megan. So moving to our third speaker, he's already coming. It's my great pleasure to introduce you, Dr. Pierre Cordelier from Toulouse. He's the director of Toulouse Cancer Research Center. For who doesn't know, it's a very unique place that they combine innovation, basic research, translational research, cancer research in a very unique place. Pierre, we're going to talk about, I hope, pancreatic cancer. He's a very important researcher and award for this right now. Thank you very much.
Thank you very much, Leandro, for the nice introduction, the kind invitation. And we'll extend those acknowledgements to Bruna and Alexandre for the perfect organization. And it's a real pleasure to be here today talking about our research on pancreatic cancer at Toulouse. So if you don't know the place, you are more than welcome to come to our place. This is what we call a comprehensive cancer center, when you can have a clinical department dedicated to oncology on the side and a cancer research center on the other side. So we are more than 420 people in this center, only for the research part, and more than 1,200 in the clinical part. And you see that's a pretty nice campus. Leandro, you had the opportunity to visit it, Vitor in the room also, so say it's a nice place. With the companies, downtown Toulouse is a very nice place to live and to work, especially if you like seeing big planes and playing rugby. And we have a connection with CROs on site. So why I'm here today? Basically, because we have already worked together with Ribeirão Preto Medical School in terms of the summer school 14. That's now organized in the next 10 days, I think, in Ribeirão Preto. Vitor and Leandro have been strongly involved in this organization. So student exchanges, it's always good to start with students, and then we can build funding opportunities. It's very interesting to do that. And we have a modus of operandum with the University of São Paulo, I think, with the Ribeirão Preto Medical School, with our university in France. So it's a care program. It's a graduate school that is interested in cancer, of course, rejuvenation and aging. So here also, there are more opportunities for student exchanges. All right, so my team has been dedicated to work on pancreatic cancer about 20 years from now. And you know, it's a very nasty disease. 1 person out of 64 will die from this disease. That's a worldwide numbers. This is very sad significance because almost everyone is dying from cancer on the pancreas, and most of the time there is only a year or so between the diagnosis and the death of the patient. That will soon become the second leading cause of cancer deaths, even if it's not the most prevalent cancer in the world, far away from lung cancer or colorectal cancer. And we obviously need effective therapies to try to move these basic findings to the patient bed. So in my group, we are organized in 3 main projects. One is to identify mechanism of resistance of pancreatic cancer in pancreatic cancer tumors, then to try to intercept those mechanisms with targeting protein interaction with intracellular nanobodies. And then I'm more into going for intervention in the clinical side. So we started a long time ago for gene therapy to try to defeat pancreatic cancer resistance to chemotherapy, especially gemcitabine. So we identified three genes that were downregulated in those tumors. So when we put them back in animal models, then in patients in phase 1 and 2, we had this kind of chemosensitization, so it was doing well. And this is an example of what you can find. Here it's a tumor, it's an echoendoscopy because those patients will not get surgery because the tumor is too advanced. So we are treating the primary tumor. You can see the needle of the endoscope, and that's a gene therapy product delivery, and then you add the chemotherapy on it. And for some patients, we had very interesting responses when we combined gene therapy and chemotherapy. This is a patient after 2 months of treatment, and he was doing well. But still, we are facing limitation, and we done that in nearly 100 of patients in Toulouse first, and then with other friends in the French territory. It seems to be safe, feasible, so that's opened a pipeline for basic research findings to reach the clinical bed, but we have a limited efficacy because we are using non-viral vectors. And that's why I got interested in oncolytic viruses that we'll talk about today. Are very interesting because they can infect normal cells, but they be blocked in their replication by, we heard about CGAS and STING, and also the MARV system that will detect the viral genome into infected cells, so it will put them in pause or even die for the community. So the virus will not be able to replicate and to spread to the tumor. Honestly, this is what I was very interested in, that the fact that the tumor cell becomes a factory for virions that can spread from cell to cell and then progress into a population. And of course, you can reprogram the microenvironment of those tumors with alarm signals that will recruit immune cells, and then in combination with immunotherapies may help the tumor to go down, but it's not immuno-oncology, so I will talk only on serious science, basic science, just like Leandro said. So let's focus on those viruses. They have hard time to deliver, and it's something that I took from innovation. You have visibility over time, so you have a technological trigger. Everybody is very excited about a new technology, a new approach. Then you have way more expectations than you can offer, and then you are completely sad. And then you're starting to think about what you could have done separately and differently, and you're starting to reach a plateau of productivity. And I think we are almost there, speaking of the field of oncolytic viruses. And so today I will talk about two things, some unpublished results on how to identify host factors for oncolytic viruses in primary cultures from patients, so that will help select the patient that might benefit the most from the therapy, and then how we can improve moving those viruses to the clinical side, because by doing some unique preclinical platform we are starting to put together in Toulouse. So I'm not focused on one type of virus. I'm very interested about the crosstalk in between the virus and the tumor cell, because they are way different. They can be different in size, in genome, and in structure and everything, but they have to benefit from the tumor. They are obligatory parasites, alloparasites. There are no bacteria. They have to feed from the tumor cells, and that's what it is very interesting. So this work, we talk about one of the interesting parvovirus, H1PV. Why we are working on it? Because it's not a human pathogen. We don't want to induce a disease when treating patients. It's highly sensitive to interferon signaling. So if the signaling is still there in normal cells, that will block virus replication, as I just showed before, and especially that will give you a first trigger for the virus to do its job in a cancer cell. It's been shown to be oncolytic in multiple cancer models and safe in clinical trials, because there have been one phase 1-2 clinical trial that show no serious adverse events at all. But we found that these viruses shows diversity in infection efficacy in pancreatic models. That's stands for PDAC. So if you have a range of sensitivity towards resistance, you can put together all cell lines, HPD being a normal cell line. It's really resistant to the infection. And we added more translational value to this work. And all those cultures, starting with the PDAC, and number, and T, are primary cultures that have been isolated from patients. And you see that there is a diversity of response to the virus. So we need to know better how the virus is getting into those cells to be infected and then efficient. There's been a lot of work on this virus, on different models, mainly since the 60s by a German group. And there are partners that have been identified. So first, it looks like some anchoring protein, just like laminin B1, that has to be highly glycosylated. You see that glycosylation, sialylation is something very important for cells and for proteins to interact with viruses. And it's been involved with sialic acid and O-glycosylation on this laminin. That's for anchoring, that is known. So this is a viral particle here in purple. It's way out of scale because it's only 25 nanometers. It's a very tiny virus with no envelope at all. Then the galectin-1 is involved in internalization because it's interacting not with the virus, but with the sialic acid motifs in the laminin B1, and then it's entering using the clathrin coat system with mediated endocytosis. But so far, there is no receptor for H1PV that has been discovered so far. So what we've done is to go for a whole genome CRISPR-Cas9 screen. So we took one of those primary cultures because we want to have a high heterogeneity. We know that those cells are not heterogeneous, so it's very good because it's kind of mimicking what you can find in the patient tumor. And then we were expressing with an lentiviral transduction one small guide RNA against one gene. And then they were infected by the virus, and we sequenced what was before and after to know exactly what the virus could help getting into those cells. Because here, if the gene of interest is knocked down, the cell will survive. And this is the result of the primary screen. And you can see that it's a color code. Here we can find some proteins are involved in sialic acid metabolism. That was expected. Vesicular trafficking, protein secretions, and others. So at first we were not so very happy. We were just, okay, let's back to the textbooks, but that's okay. That's research. Good to see that you can find stuff that has been thought about 20 years ago. But then we went to a secondary screen, meaning that we went for transient CRISPR-Cas9, not to modify the genome of those cells, same primary cells from patients. And then we have interesting surprises. So if the bar is going up, it means that knocking down the gene will protect the cells from being infected, meaning that they are super important for infection. It's a mirror way of thinking. And we had those proteins, and you see here that some genes were not validated. This one for sure was not. This one has opposite effect. And this one drew our attention. Why TGOLN2? Actually, that's the name of the gene, but it encodes for a protein that is named TGN46. And this is an interesting protein because it has some luminal part. It's a not so big protein, but it has a luminal part, and then that can cycle to the membrane of the cells. So it could be a good candidate being a receptor. So basically, it's cycling from TGN means trans-Golgi network. So I mean, this is protein that is everywhere in the trans-Golgi network. This is here, so it's a representation of TGN46. And then it's known to be important to the export of specific cargoes named CART that are overexpressed and overrepresented in pancreatic cancer cell lines. So it's quite interesting that it's quite specific to pancreatic cancer. And what is not completely sure of is the proportion of the protein. Most of it, you will find it in the cytoplasm of cells, but a fraction of it will recycle to the membrane. So we thought that might be a good candidate for being a receptor for H1PV. So we went for loss of function and gain of function experiments. So I will, for the sake of time, I will show only loss of function and sometimes gain of function. So see here in green, those are the cell lines that are knocked out for the protein. And so if you infect them with high MOI, MOI is the number of infectious particle per cell. And if you go for 100 MOI, it's a huge, you're literally pouring virus on top of the cells. So you have a really nice protection in terms of viability and then in cell counting. So you cannot kill the cells anymore. Why that? Because you don't find any genome or very few of it in cells. That was done by qPCR. And this is something that is dose dependent. Again, that's very interesting. And of course, you are not seeing the virions coming out of the cells. So there are less production of the virus in cells that are knocked out for TGN46. And that's the same, it's a little bit small, sorry about that, but it's the same for viral proteins because we looked for NS1, which is one of the 4 proteins encoded by the virus. It's easy to to study when you have these small genomes. And this one is basically doing everything in the infected cells. So it's going down when you knock down this protein. And we have also access to an antibody that is specific of capsids. So capsids means assembled in the cell. So that's it is a way to check for cells that are producing mature variants and see the numbers are going down. The next thing that we put together was an internalization assay by qPCR. So here in green again, if you let the virus on top of the cells at 4 degrees, so endocytosis is blocked, and then you release at 37 degrees and you do PCR at very early time points as compared to the results I just showed you before, you will see less viral genomes and more if you overexpress the protein. And there we go for this release using immunofluorescence. So in green, this is TGN46 that's showing up. In red, that's a viral capsid, and EEA1 is one of the partner in the endosomes. And if you look at the higher resolution, you see that there is a perfect match in between TGN46, the viral, bless you, the viral proteins, and EEA1 in those cells. If we try to, I told you that TGN46 is recycling a lot in the cells, so it's really hard to see that at the membrane. So we had to block all the exocytic process using a drug that I love. It's dinosaur. Everybody walk the dinosaur. And so you pretreat with this drug. You incubate with high numbers of virus, and then you release and you see what's going on. So we set up the dose with transferrin, which is transported by this endocytosis pathway. And now what you can see is that most of the virus and the protein are colocalizing at the cell membrane. The EA1 is not anymore. That makes sense because the virus is not inside. And if you look deeper, you have this really nice colocalization at the membrane of the infected cells. We went further trying to figure out how it works. So we went for modeling. And there was this assumption that in yellow here, that's a cellulation pocket from the first domain of the protein, transmembrane, this extracellular domain number 1, that was of our best guess for interaction between the protein and the virus capsid. And so we did some molecular biology to cut and remove some pieces, bits and pieces. And very quickly, what we found is that if we overexpress the protein, once again, we have more virus entering into the cells. That's a positive control. If we express GFP as a control, nothing happens. This is very funny, but there is an explanation. You always find the explanation after doing the experiment. I mean, we are scientists. If you put a flag on the N-terminus of the TGN46, nothing happens, and we were, something is not working properly. If you put that in the C-terminal end, in the part in the domain that is inside the protein, still you can rescue in those cell lines that we have previously knocked out for the protein. If you try by expressing others mutant of the domain 2 and 3 here, we made sure that they were addressed at the membrane, but it looks like it's not doing anything. But here, if you cut out the domain number 1, the virus is not able to enter the cells anymore. And it makes sense because the flag is changing. I mean, the conformation of the domain 1 is masking the domain 1. So here it makes complete sense once you get the answer, of course. This is not the way we are saying the story, telling the story in the paper. So just to wrap up with this part, maybe we had something quite important to know better which patient to treat, what patient to treat with what type of virus. So still, I think that laminin is very important. Galactin-1 is more stabilization than anything because those viruses need to tether before getting into the cells. It's predicted that you need 10,000 particles to get in one cell for one infection. So it's kind of a lazy virus anyway. And now, oh, sorry, it's a conversion probably. The binding with TGN46 here with the N-terminal part seems to be mandatory for virus entry. And then, of course, it's Clathrin-mediated endocytosis. So what will be the translational potential of that? So we did all those tests of virus infection in normal cell lines. So here, they don't care at all. IC50 of infinity is very interesting. But these are cell lines, and we did the same for primary cell culture. I don't think that by itself TGN46 will drive the infection, but you have to consider what is known. Because if we put together the cell lines in the range of sensitive to resistant, you have different level of expression. See already the difference in cellulation might be important. But if you match with what are the already known predictors of H1PV infection, you see that laminin B1 is going clearly on the left, galactin-1 also. So it seems that you need a combination of all of those. And we found also the enzyme that is responsible for cellulation, specifically cellulation of the domain 1, might be also part of this thing. And this is quite interesting because it seems to be a key lock system on-off system, because the normal cell in which you don't have a particle, we worked hard to find the virus particle in those cells. The only marker that is absent from that is TGN46. So I think this is more qualitative than quantitative, especially because we have to deal with glycosylation. What we are doing now, we are trying to move that to clinical applications. So we heard about organoids and primary cultures. We have a nice collection of PDX and the largest cohort of patients, so we can benchmark the signature and make it round and refine it more and more. We can work with friends in Marseille on a really well-developed collection of organoids and primary cultures, and we've worked on a signature, on a score, and we are currently now performing some tests about is the red one or the one that gets more infected by the virus. And I will take the last 2 minutes to share with you what we might do in the future to prop up the field of oncolytic viruses in clinics. I mean, there is a strong attrition, just like for any innovative therapy, between the number of tests that have been done in preclinical setting or in phase 1, with only 1, maybe 2 now approved strain by the FDA and EMA. So we partner with a company that named Neovirtec, which job is to reveal viral DNA replication with the OnCOS system. And this is very friendly user, because I don't know, for those working, I'm not a biologist, I'm a cancer biologist. But the thing is, when you have to title your virus with plaques, it can be super annoying, especially when it's late, when it's getting late during the day. But here the plaque is showing up green. It's not because the virus is expressing a protein, anybody can do that. But because the viral genome is tagged with a system that makes it fluorescent. And that's even more interesting when you have this kind of, I don't know how to, so I think to do better in Portuguese, English, Portuguese. So how can I remove this? Yes. Yeah, but I'm not very familiar with the options here. All right, thank you. So I'm going to click here. Sorry about that.
Not going.
I think you need to remove the pointer. He doesn't want to go. Yes. Thank you, Leandro. So here what you see moving are viral ribosomes in live infected cells. So you can have a real output of what's going on in cells at the cellular resolution. And of course, we benchmark this system with spheroids and now working on organoids. And you can have a very quick answer whether a tumor population will allow or not replication. And then you can go inside the spheroid with light-shift imaging. So purple here are infected cells and blue is DAPI. So we are going from front to the outside of the spheroid, and we are applying a AI-based analysis of viral spreading. So on the left with a nice color is the convolution of the cells with different positions in cell cycle, and then on the left, on the right, sorry, it's a model of viral replication that we are working with computer scientists that are hosted on site in the CRCT. And we were recently awarded by the Bank for Innovation and the Plan France Relance. as long as it stays. We'll see with the next election. To make a platform with different partners, from cloning to producing those viruses to test for anti-cancer activity in screening, optimization, as we can optimize those viruses by directed evolution or combination with other drugs that are already on the market. We heard about that this morning. I'm more involved in the part using tumor organoids and safety on normal organoids, and then we have this image analysis and in vivo studies. And we are pushing very hard now in Toulouse to connect with friends from last CNRS. I think I leave you, it rings a bell somewhere. And to prop all this organ-on-chip system, because in there you can work on real tissue samples, and you can explore the role of the immune system and how the virus can get rid of the stroma, which is quite a huge deal in pancreatic tumors. And so I will stop here. And thanks again, FAPESP, for the very kind invitation. Don't forget that we have already stuff together with a, yes, now I'm master of the scene, with the SSM 14. I will be in 2 weeks, but then, you see Toulouse somewhere. So in 2 years now, you're more than welcome to attend to the one that will be in Toulouse. The care system, all our friends. Marseille for the PDAC resources, IMCOR, which is Russian and Tech Network, we've been sponsored by them, and the ARC, which is a charity in France that is supporting our work, and all the people from the team and clinicians that are helping on a daily basis, and of course the patients. Thank you very much for your attention.
So last but not least, we're going to go with the talk of Professor José Barreira Cavalheira. He's a full professor of oncology at UniFesp, and recently he was appointed the head of the coordinator of oncology in the entire country in the Ministry of Health. Thank you very much for taking this position.
And I increase my responsibility here, and I'm going to change everything because I'm not going to focus on the cancer. I'm going to focus on the patient with cancer. And then I'm going from obesity to cachexia, and then try to explain this pathway. Let's go. Oh. Foi? Okay. So that's my agenda. I'm going to talk about obesity and cancer, then contextualization, metformin, then cancer cachexia, new contextualization, anorexia, increased energy expenditure, muscular changes, adipose tissue changes, and the conclusion perspectives. Then, just to talk about the pancreatic cancer here, Luciano Pavarotti is an obese person that died from pancreatic cancer, and this is the numbers for obesity. Then you have here in 2020, 9.5 million, and it's expected for 2040, 4,009 million of persons, both with just because of demographic changes, but also because of risk changes. And these persons with demographic changes, it's mainly in middle income countries more than in high income, because they have also changed the epidemiology there, and you are changing faster here. And then these are all the cancer associated with obesity and overweight. Then to have all these cancers from multiple myeloma in blood to meningioma in brain, then it goes everywhere. We need to have some things in common. And what we can see is that obesity in the center, diet on one side, physical exercise on another. Then we have the mechanism with growth factors, insulin resistance, inflammation, and adipokines, steroid hormones. And then the biomarkers here is IGF-1, insulin, leptin, adiponectin, TNF-alpha, estrogen, progesterone, and the cancer. Then we have growth factors on one side, and then inflammation on the other side, and then we have a conception. And with this in mind, we try to see something about a drug that was in that center. Then we try to work about 10 years ago, but it's an interesting study that we use metformin with Taxol, and we see a synergistic effect. Then in a drug that treats insulin resistance could increase the effects of Taxol. And this was because of proliferation effect that you can see here. I'm going to show that this was because of metformin and Taxol synergistic way to decrease proliferation. This was a highlight and at that time, and also the tunnel effect in the apoptosis. This is what I would, at that time we showed that this was a bioinhibitory effect. We know that there is a lot of more effects than that, but what I'm going to show here is that after almost 10 years, we could do a phase 2 study with metformin and irinotecan, and we could show that there is patients that were that received multiple treatments, they still have an effect that's similar to the effect that you see with the approved drugs. This is a point that needs to be thinked, because this is a drug that it's for middle-income countries, and you can't see the phase 3 drugs of this because there is no interest for pharma. Then it's a point that should be discussed here at FAPESP. And then change the gears to cancer cachexia. That's the fact is this is a patient for mine, and for many years we didn't see obesity as a cause of cancer because of they came to the clinics like this one that is now cachectic. And we worked with cachexia, and these are the bases of cachexia. That here is the tumor, TNF alpha, IL-6, IL-1, interferon gamma, the inflammatory effect. I think that the mouse is there. Okay. And then we can see that it's not working. Okay, now. Then we have here for each center, cachexia is not it's a multi-organ. If there has a multi-organ effects that it goes to the brain, to the skeletal muscle, and white adipose tissue, and then we have here an effect together with ghrelin and PTH-RP to go to anorexia, with PIF and activin to go to MRF and atrogene, decrease and ZAG to the white adipose tissue. This is going to lead to reduction in food intake, increase in energy expenditure, reduction in muscle tissue, reduction in physical activity, reduction in adipose tissue. Then we work in every aspect of these in these intersections. And here is the definition of anorexia. We have cytokine hormones, nitric oxide, that's going to activate in hypothalamic tissue and cause malnutrition, cachexia, increase the morbidity and mortality, worsening of quality of life. And here we see how is it and how it works in the brain. It works in the arcuate nucleus with leptin and insulin being the most important regulators that goes to the first order neuron with the NPY and the AGRP and pro-opioid melanocortin neurons that are going to be integrated into the second order neuron that is in the paraventricular nucleus and control food intake and energy expenditure. Then here we worked at that time just to why I worked with cachexia too. Our group described the effects in obesity, how obesity become obesity. It's because one of the main facts is because of the insulin resistance and leptin resistance in hypothalamus. And we show that here that the obesity mice, they have a decreased insulin receptor activation and AKT activation in arcuate nucleus. This was the same, and then we put, then in this context, we go to the tumor and in a model of cachexia, and we could decrease the inflammation in the central nervous system, and decreasing the inflammation in the central nervous system, we could reduce the activation of INOS and reduce inflammation in the central nervous systems. The same was true for metformin, and we could do an effect of tumor plus metformin with a little increase in food intake. And that's like this in the brain, then IL-beta, arcuate nucleus, MSH, NPY, and AGRP go to food intake and increase energy expenditure. And this is what that was my contribution for this area. And going to the muscular changes, I then came back. We came from bedside to bench, and then from bench to bedside now. And working with cachexia, we use CT for ETL3 to evaluate, and we made the AI with segmentation for deep learning, and you could measure the muscle, the intramuscular adipose tissue, visceral adipose tissue, subcutaneous adipose tissue, and then we can do like that. It's the manual segmentation and our algorithm, and with this algorithm we could put together these radiomics together with the clinical and some genomics that we are working on now. Then we contribute in this area, showing initially, then we went to bedside to understand better this effect in cancer. And we contributed to know how sarcopenia decreases the prognostic is associated with the poor prognosis. Here in colorectal cancer, we saw even disease-free survival and progression-free survival. We also obtained data for myostatosis, that is the increasing adipose tissue, and we showed that myostatosis is also associated with a decrease in prognosis, with the decrease in disease-free survival and overall survival, and also in gastric cancer. Then we were at that time paving the way to understand better this. And then we did translational interdisciplinary way of thinking. We used the adipose tissue to try to see these two. The adipose tissue is characterized with inflammatory cells, like in diabetes with crown-like structures like this. And also there is a browning of adipose tissue, and they become beige when patient here is in animals with C26, it's a cancer with IL-6 proficient and IL-6 deficient, and then you see that there is no browning without IL-6. Then we tried to do this with CTs, and we delineate the subcutaneous adipose tissue in these conditions with using the radiodensity, and you could see that there is here in multiple myeloma patients, and you see low, intermediate, and high radiodensity in these patients. And you see the radiodensity is completely associated with IL-6 and with TNF-alpha. It's not so much, but it's associated, and also with that's what I'm going to work on and see what's really changing the radiodensity that is inflammatory and coming back to the bench to see this. And here is the radiodensity, and here is the survival that we put this in the literature, showing that the adipose tissue is the radiodensity is associated with survival. Here for multiple myeloma, then for colorectal cancer, and the radiodensity is also associated. And then we go on and saw if this was associated with the glucose uptake, and used the nuclear medicine with PET-CT to see if the radiodensity is associated with higher glucose uptake, and that's what happens. These are the conditions: survival according to glucose uptake in patients with multiple myeloma, and here you see the glucose captation, and this was original contribution to the literature, and then we can see the metabolic status of the patient. In a cancer patient, you're using tomography. We need now to go on this and integrate with we are collecting samples of adipose tissue, muscle tissue, and now we are going back to the bench. Summarizing, decrease in vet set area is associated with poor prognosis, decrease leptin, increase in pro-inflammatory cytokine levels, increase in adipose tissue glucose uptake, and decrease in survival, overall survival, and event-free survival. Then that's fast. Understanding the pathophysiological mechanism and the clinical implication related to cancer at extreme of obesity weight spectrum is essential to improve our efforts in cancer prevention, diagnosis, and treatment. Certainly, metformin is not the only expression of the pathophysiology pleiotropy that relates to the intersection between obesity and cancer. Modulating obesity in new treatments, such as immunotherapy, represents a promising area of research. An integrated and interdisciplinary approach is certainly opens horizons for original approaches in the evaluation of cancer cachexia. Integration with the advanced computational tools for body composition, combined with clinical and genomic data, will certainly enhance the prognostic capability of currently available tools. The use of imaging obtained not only with the FDG, but also with the other radioisotopes emerges as a new and valuable tool for the study of cancer cachexia. Now, what I'm saying is that staging not only the cancer, but staging also the patient is very important, and it probably explain a lot of things that we are not understanding, and this is an approach that you can understand better the patient. And then to finalize, I would like to acknowledge the patients and all these people that work in this condition. Muito obrigado. Merci beaucoup. That's it.
I'm going to invite the four speakers. We are a little bit late, but we have time for one or two questions from the audience. If Marion, Miriam, and Pierre, thank you very much. Why, please, how?
Hello, a very nice presentation. So I have a very quick question about what you presented on the deep learning aspect of segmentation. Is it already in practice? Do you have it implemented in clinic? And what are the, for example, similarity index? Could you say, like in percentage, what does it give?
99% is the groundbreaking, and we can reproduce all, but we are using without putting clinics. We are using just in research. Or you can say again.
Yeah. So when you say 99%, so I suppose you're comparing with the ground truth, and the ground truth is a human ground truth.
Yeah.
That means a radiological ground truth, like you showed the CT and MRI scans. So how could you expect a 99% of ground truth of AI with respect to human, which is anyway vulnerable to errors? So how would you implement a 99% replication of human, but we are not sure that humans are 99% of
No,
times, right?
but it's about the muscle adipose tissue. It's very fixed, and then we have done with about 300 patients that we have done manual segmentation. Then we go to the ground break. The only one that's smaller is the adipose tissue, intramuscular adipose tissue. All others are up to 95%. It's very, very, very close to the intramuscular adipose tissue that's very small. I think that the manual segmentation is worse. That's what happens, because when we see the image, we can't see almost any difference. This was done for gastric cancer, the cohort of gastric cancer. Then we are expanding now for a cohort of hepatocarcinoma and for a cohort of with collaboration that we are going to increase the patients to about 400 patients, but we are not using in clinicals yet. Yeah.
Great.
Any other questions? Go ahead, Chab.
Hi, Marion. Great talk. I'm a great enthusiast of your work, of your group. I've been working with something related to that, and I've been wondering how you have classified the stamens in CAR T cells, and particularly if there is any key transcription factor involved with the induction of this profile that goes beyond BAC2. And now, so I essentially forgot if there is any pharmacological inhibitor that could target specifically SUV39H1. And why did you decide to use CRISPR instead of a pharmacological inhibitor for the clinical trial, if there is one?
Okay. Yes. So for the stemness memory gene signatures, so we took different gene signatures that were already published. So there is no one gene better than the others. So different published signatures. Three, I remember three. Then, yes, there is a pharmacological inhibitor of SUV that we have been using in the one mice paper when we do the reprogramming of SUV through and the reprogramming of the cells through anti-PD-1 plus SUV inhibition. We had both models, genetic inhibition and pharmacological inhibition. It worked less with the pharmacological inhibition. And then here we want to target specifically the CAR T cells. So this is why we're doing the genetic inhibition through CRISPR-Cas9 in order to really target the CAR T cells specifically, instead of giving an inhibitor to the patient that would not specifically target the CAR T cells.
Can I ask one more question, Leandro?
Sure.
We know that modulating the epigenome of whatever cell you're targeting can make drastic changes in the cells, right? I was just wondering, when you target SUV39H1, you have seen any differences in the activation of, for instance, of an oncogene or the inhibition of tumor suppressor gene in a CAR T cell or something like that?
No,
something that could be harmful for the CAR T cells.
Yes, we haven't seen this. So this is one way for us to argue that it could be a safe option. We haven't seen this.
Thank you.
Unfortunately, we are running late, and we have some. We have to finish here. Sorry. So, but tomorrow we're gonna restart at 9 o'clock. Thank you very much, everyone, for being here until 6 o'clock. Thank you one more time for the presenters, amazing presentations and data. And we see you all tomorrow. Thank you very much. For the presenters from France, they're going to go hotel. Please wait here so we go with the van together, okay?